Welcome to BrainLink X Lab Day talk concert. Good afternoon. I'm Sang-gu from Konkuk University. I'm the organizer of this event. With the support from the COPST, I really thank you for all the supporting from the COPST. So, through this BrainLink Global Technology Exchange Project, world-renowned researchers collaborate with domestic young and mid-career researchers to share cutting-edge research results and promote the international joint research. So I'm the domestic organizer, and Dr. Kim from Johns Hopkins University, he is now coming. He will arrive in 20 minutes. This event is held for 4 days and 3 nights from July 9th to 12th on the theme of Convergence of One Health, Advanced Regenerative Medicine, and Artificial Intelligence. So on the first day, today, we have the talk concert with 8 the world-renowned speakers: Luke Lee from Harvard University, Andrew Packard from Johns Hopkins University, and Dr. Kim from Johns Hopkins University, and young Charles Zhang from Emory University, Kyun Lim from University of Sydney, Australia, and Xian Lee from Stanford University, and Saskia Schuker-Big, right? Okay. She's from Utrecht University in Netherlands, and Michael Cho from Iowa State University. So after this, the talk concert from July 10th to 12th, we'll have another event which will be held in Venezia Hotel in Yeosu. So in this event, these 8 foreign speakers and also the 18 professors from domestic university, such as Konkuk University, KAIST, Korea University, Busan University, and Suncheon University, and also the 16 young researchers from Konkuk University, KAIST, York University in Canada, and Johns Hopkins University in USA. So from tomorrow we'll have the BrainLink X Lab Day event. So it will be held in Yeosu, and at this event we'll have the 4 sessions. Session 1, cutting edge advance in disease modeling and therapeutics. Session 2 is the frontiers in biomedical innovation, functional peptides, stem cells, and EV-based therapies. And session 2-2, frontiers in biomedical innovations, AI-assisted platforms and genomics. And the last session 3, the cutting edge advance in deciphering cellular signaling mechanisms. So this is the detailed program. So you can check this detailed program from these books. Okay, interested in this book. This is session 2 and session 2-2, and this is session 3. And today we have the talk concert with 14 excellent speakers. So for this concert, I will moderate, and also the Dr. Ahmed Abdal Dayman from Egypt will moderate the talk concert. Will you have big welcoming remarks? Okay.
Good afternoon, everyone. Welcome to BrainLink 2024. So it is our privilege and honor to welcome all of you. Especially today, we have very, very special and prestigious scientists in the field of virus and organoid and exosome and a lot of interesting things. So please enjoy today's talks and try to make questions after the end of the session. And welcome. Thank you. Please join me to give big hand to this outstanding speaker. Thank you.
Okay, thank you, Ahmed. Okay, so now we will start today's talk concert presentations. The first speaker is Luku Pangse Lee. Pangse Lee is from the Harvard University Professor of Medicine, and he is also the member of Royal Society of Chemistry and American Institute of Medical and Biological Engineering. His research focuses on the quantum biological electron transfer in living organisms and early detection in infectious and neurodegenerative diseases. Today he will talk, the topic of today's talk is brain organoid map for neurodegenerative diseases. Professor Luke Lee, please.
Thank you. It is my honor to be here and share. Please forgive me that I might have to skip many slides because I thought this is 45 minutes. I never informed that it was 15 minutes, so I'll just try to my best. First, I'd like to just introduce that our lab is a poet. We are trying to write a poem, Patient-Oriented Engineering Medicine on Chip. That's our goal. We really like to really embrace the poem so that we can really continue to write technological poem on a chip for the diagnostic as well as organoid on a chip. But briefly, I'll introduce you some other background before I move on to brain organoid so that you get a better idea of what we are doing. So SmartSend for us is integrated molecular diagnostic for global health. Emphasis is sample to answer a device at low cost. So we've been working on integrated molecular diagnostic chip. And this is more than 10 years ago, we developed this chip. And then during the COVID, my student made a company for this Lucira and made the first FDA-approved home diagnostic chip. So this is the first home diagnostic point-of-care system that we developed for nucleic acid RNA detection, not the protein detection. And then there's a slightly different design, but make an automation so that you can have a multiple detection of the nucleic acid so that you can have a automation of a sample prep. There is no pumping because there is a vacuum battery is integrated inside of this chip, so there is no tubing. So it's a simple and effective way of making digital sample detection so that you can have a nice blood separation or other biological separation so that you can take advantage of detecting RNA or DNA detection for different infectious disease. So we are still ongoing to detect both protein marker and nucleic acid marker so that our goal is really executing multiple detection for environment as well as our body, as well as water and so on. So this is our focus, but we like to show you that how I like to encourage many of students that it's your hand to take care of our body and earth as well as making new invention. So this is your time to create multiple invention, but due to the time, I like to just highlight a few things out of many different development of our device. But the soul is a single cell to organoid to understand life. So a long time ago, this is Dino Di Carlo. He was my student, undergraduate student and PhD student. Now he's a chairman of UCLA and work on this single cell trapping side so that you can understand the enzyme activity after you trap the single cell. But then we worked on the physiologically relevant dynamic 3D cell culture chip. We never called lab on a chip, organ on a chip. It was even before organ on a chip. So this is early 2006. But I'd like to just highlight the Julius Petri invented the Petri dish. You might laugh that, oh, is it really invention? Yes, this is invention back then. This is an important invention so that his boss, Professor Hans Koch, get a Nobel Prize. Okay, but this is excellent for cell culture for something that you don't have to use dynamic flow. This is excellent, but you are saying that we are still thinking about that Earth is not rotating. Earth is flat. Earth is rounded. There's a dynamic flow. So we have to think about making new system that allow to make dynamic flow and perfusion control and without statics, so that you can mimic circulatory flow as well as interstitial flow. This is tissue sample. There's a circulatory flow, interstitial flow, so that you can create a similar environment, microenvironment in the chip, so that you can have a multiple experiment. So I'd like to encourage you to young student. This is again another my student made company out of this chip, so that the first actually 3D cell culture chip, and then purchased, which transferred to Millipore. But the first one that I introduced for the diagnostic chip was transferred to Pfizer. Okay, so organoid. So let's talk about organoid before I go to brain organoid. This is a wonderful demonstration by Lancaster. Everybody really get excited. I was excited, even though I'm not an organoid person, but I decided to challenge to work on organoid. So again, they start with a static condition. You know that our cell, organ, supposed to expose physiological dynamic flow. Earth is not flat. Earth is rotating. But anyway, so they made a nice demonstration, but it's a different stage. I was thinking, because we have to think about purpose-driven science and technology. If you really want to make drug screening chip, this has a problem because it's always different stage. It's not the same condition. So we have to solve the problem of this non-uniformity, and also we have to think about limited monitoring and the screening and so on. So I'll skip many details, but the purpose of the MAP, MAP is microphysiological analysis platform. As you can see, there is a dynamic flow and recapitulating physiology and so on. So first, due to the time, I'm going to skip this detail of the chip, many different neurodegenerative chip. I'll skip this, but I'll move on to directly the brain organoid chip, brain organoid map. So this is the first time we didn't publish this yet. It's on being reviewed, but I decided to share with you because I trust you. And then this is a really exciting time to think about real-time, non-invasive monitoring of neuropathogenesis dynamics or drug discovery through this system. So this is the system that allows to capture brainwave as well as detect collecting exosome. So this system has the dome shape to form the brain organoid, as well as two different channels. Middle, the blue channel is forming the organoid, and then red channel deliver different factors. So you can do many different systematic study due to the transcription factor or many inflammation factor and epigenetic factor, and so you can perturbate system. So at the end, the result is this. You can see this uniform organoid because it allowed to control the flow, and then you can create very uniform size of the organoid and same stage. And not only creating uniform size like this, but also this is a cross-sectional view. You can collect exosome from organoid, and then you can detect brain wave without touching cell. Many people are misunderstanding, unfortunately, even neuroscientists. I'm not a neuroscientist by training, but I'm a biophysicist, but they touch the cell on top of the MEA. MEA is measuring local electropotential and measuring brain wave. So this is important to think about putting the electrode on top of it. Our EEG is not touching our cell, right? We are measuring the EEG on top of the skull. So this is electrode design, and then we improve the noise, and then this is a normal brain, mini brain, and then you can see if you perturbate it with the inflammation factor, you see the beta wave change and the gamma wave change, so you can monitor non-invasively in real time. Also, if you use a PTG, and then you want to see how the epileptic behavior change, in this case showing that if you put too much drug, you cause more epileptic. So you have to find best drug concentration and amount and so on. For individual cases different. So I'm going to skip this one. But this is ongoing, our project. We are making a Parkinson's disease model so that we can compare with the Parkinson's disease organoid with the brain wave, as well as we are collecting, as I mentioned, exosome, and then make sure that we can take advantage of both molecular expression and physiological expression so that we can really understand. So we are capturing the exosomes from this system and compare a healthy and Parkinson's disease model. I think this is same. I'll skip this profiling, and then we can capture and characterize this mini brain organoid, and then we can also capture the calcium wave. But this is my favorite part, that you can really capture many different brain waves from this Parkinson's disease. It's an IPS. I was very surprised to see when we really model this Parkinson's disease organoid and capture similar behavior of the brain wave. And this shows that many different times you can observe why delta wave is generating more excitement. And we can also test with the MPP plus and see how this MPP plus is behaving, organoid is behaving due to the neurotoxin. So as you can see, if you deliver, then you will see the different, change of the brain wave that you can detect. And then also you can use if you understand 1F noise of the brain, the neural network, you can also understand, you can take advantage of the understanding 1F noise to see how neuron to neuron is interacted. I'd like to just, I have a few more minutes. So, how many people think that neuron is dominating our brain? Everybody, right? I was thinking like that. You know, percentage of the neuron in our brain? It's only 20%, not 90%. In our society, there's a star, but majority of people are supporting the star. The neurons are star, but that doesn't mean that only neuron is making network and generating brain wave. There are many, many supporters, especially astrocytes, glial cell. They are playing an important role even in oligodendrocyte. If I have time, I could really explain this molecular pacemaker. You know the pacemaker here, right? You can think about tiny, tiny nanostructure as a pacemaker for the molecular diagnostic, especially mitochondria. But can you hear me? Okay. All right. So I guess in 1 minute, I'd like to highlight why it is important to understand quantum biological electron tunneling in life science. In the mitochondria, there's an electron transfer chain. If you can modulate, if you understand how electron transfer happen in that membrane, then you have a lot of the new insight, and you can create new invention for this. So due to the time, I'll skip this result, but I'd like to just show you that we can measure oxidation reduction of the cytochrome in mitochondria optically due to the quantum biological electron transfer. Imagine that if you can measure, we can modulate mitochondria to rejuvenate. You are talking about regeneration. A lot of the disease is due to the mitochondria because the mitochondria is our batteries, the cell's battery. So we have to think about how to regenerate mitochondria. So I'll skip this one. But since you are interested in a brain chip, you can really link lung and brain BBB and then understand how infectious disease can attack the lung barrier, but it doesn't really affect directly to the BBB and so on. Or you can also study gut-brain axis, and then again, you can really generate a detailed study, but due to the time, I'll have to skip. But hopefully, I give you one more exodus. This is my favorite one, how we can collect exosome more effectively using this exodus. Exodus is nothing but exosome detection via ultra-fast purification system. As you know, the exosome is everywhere, even in the brain organoid or from our even breeding and the plant and fish. But this system allowed to purify the exosome so that you have a better way of detecting the exosome. I'll skip this. So this system allowed to have a high yield and high purity, and that allowed to make a better profiling of the cancer biomarker. But I'd like to finish with this. Give me a 30-second exodus for the tears. People think that tears is just tears, but you can collect the tears and then separate the the exosome. Then if you really analyze this detail, it's really fascinating that this is the data using the human protein atlas and then compare with the exosome that you collect from tears. Then you can really detect many different parts of our body. Tears is not only detecting eye disease only because our exosome is circulating in our body because there are immune system that you can really take advantage of it. So EB detection is very important for the cancer as well as many different diseases. But then if you are thinking about EB as a therapeutic, you can really also integrate fusion and targeting moiety so that you can take advantage of this beautiful living nanoparticle of the exosome by decorating with the target moiety and fusion to execute your system. So I'll finish this with summary that convergence of the physical science and life science, engineering and medicine really need active learning spirit, and multidisciplinary teamwork really need humble spirit. And then innovative design need creative spirit, and nano microtechnology need precision spirit, and standardization need intentional spirit. Thank you.
So after all the presentation, we will have the discussion and Q&A times later. Okay, the next speaker is Andrew Pecos from Johns Hopkins University. He's a professor of molecular microbiology and immunology at the Bloomberg School of Public Health at Johns Hopkins University. And he's also co-director of the Johns Hopkins Center of Excellence for Influenza Research Response, and director of the Center for Emerging Virus and Infectious Disease at the university. And he is a specialist in the biology. He investigated how virus interact with the respiratory epithelium. Today's topic is virus surveillance and identification of novel genetic mutation that facilitate virus circulation. Dr. Pecos, please.
All right, thank you very much for that kind introduction, and it's a great pleasure to be here to talk to you about some of the work that's going on in my laboratory. I hope you can see the mouse up there too. I'm going to start by trying to tell you a story about how we're utilizing multiple aspects of primary cell cultures, machine learning, and artificial intelligence to try to understand the contribution of very specific mutations in viruses to overall disease potential and transmission in the population. First, want to thank the members of my laboratory. I've highlighted in dark here the members who contributed directly to this project. This is a project that started before the pandemic, was put on hold because of the pandemic, and then we were able to pick it up and finish it up in the last couple of years through the efforts of many people in the laboratory, while they were also spending a lot of time working on SARS-CoV-2 and the COVID-19 pandemic. And again, thanks to all the funding institutions, the work I'll talk about today is primarily through these centers of excellence in research and surveillance and the new iteration of called SEER. We're entering our 9th year of continuous funding in this program. Let me give you a 1-minute introduction to influenza virus. Please don't tell my students that I told you everything you need to know about influenza in 1 minute. But from our talk, there's a couple things that's really important to understand. First is that influenza is a negative-stranded RNA virus. RNA viruses introduce mutations into their genome at a much higher rate than DNA viruses. Influenza also has another strategy to introduce diversity into its genome, and that's the fact that its genome is segmented, meaning that each of the 8 primary genes are replicated on separate pieces of RNA. And what that means is when 2 viruses infect the same cells, they can exchange genetic material and therefore speed up the process of evolution much more quickly, because now you can move whole gene segments with mutations into new virus particles. The 2 proteins that I'm going to talk about today are, maybe I'll do this first. We'll get the pointer here. The hemagglutinin protein, or HA, which is the protein that's responsible for attachment and fusion of the viral lipid envelope to the host cell. Antibodies to the HA protein are a great correlate of protection from influenza, and the vaccines for influenza introduce these antibodies. The neuraminidase protein is another protein on the surface of the virus, shown here in yellow. It's less studied than the influenza HA protein, but is very important in terms of facilitating virus release, preventing virus aggregation, allowing the virus to penetrate through the mucus on your respiratory tract. And it's also known that antibodies to the NA protein can inhibit virus replication and protect you from infection. Finally, I'll talk a little bit about the sequence that I'm talking about. The subtypes of influenza, which I'll be referring to as H3 and N2 for hemagglutinin 3 and neuraminidase 2 in this talk, represent the larger relationships between viruses. Minor relationships are focused on clades. These are focused on particularly the HA protein, and are another way for us to keep track of mutations specifically in the HA protein. Now, our SEER Center was really put together to combine population-based surveillance for respiratory disease, put in place diagnostic testing that was rapid and accurate so that we can identify particular viruses that are driving increases in respiratory illness. We then combined genetic analysis and bioinformatics to try to identify unique mutations in the viruses that might be driving surges in cases or surges in severe disease. We then bring those viruses into the laboratory, utilize clinical isolates or infectious clone technology to characterize viruses in primary cell cultures for their replication. We also have blood from the people who are infected with these viruses, so we can study both antibody as well as immune responses, cellular immune responses to infection. And all of this helps us feed into our public health response to infection by allowing us to more quickly implement public health measures in response to a respiratory virus outbreak. All of this was established with seasonal influenza, and we also use this in our COVID-19 response at Johns Hopkins University. Now, my laboratory, the real workhorse in my laboratory are primary differentiated human nasal epithelial cell cultures, or HNECs. These are derived directly from human tissue samples, grown in culture, and then differentiated at an air-liquid interface to mimic the respiratory tract. And you can see that over the course of 2 weeks, the cells go from a rather nondescript nature to possessing multiple different subtypes of epithelial cells in those cultures. The ciliated cells are shown here. The cells that don't have cilia produce mucus, and of course, you can see these cilia beating in the culture tube, and they represent at least what we think is a very good model for the upper respiratory tract for virus infection. I'm going to quickly tell you two stories of how our culture systems have helped us identify critical mutations in the H3N2 influenza viruses that circulated over 2 consecutive seasons, 2016-17 and 2017-18. So in 2017-18, we had what we call a typical influenza season in the United States. This is the blue line here. We have influenza-like illness, a very non-specific capture of disease, and the weeks of the year on the x-axis. And you can see we had a peak influenza activity in February and March of that year, with a corresponding peak right around the Christmas and New Year's holidays in the United States. This is the one I want. We detected in that year an interesting mutation, not in the HA protein, which was the focus of the vaccine studies for that year, but in this neuraminidase protein. It was a position 245 mutation, and what struck us as being interesting is shown here in this model. The stick diagram shows you the carbohydrate that is now resulted from the creation of an N-link glycosylation site on the protein. That carbohydrate overlaps both the neuraminidase active site, shown in cyan, as well as a known protective antibody epitope in yellow that's targeted by virtually all influenza virus strains that circulate in humans. So we were interested in knowing what this carbohydrate might be doing for virus replication. In this case, we jumped right to an infectious clone technology where we could generate 2 viruses that differ by only 1 amino acid change from each other, and that's this 245 glycosylation site. So in red, we have glycosylation negative. In blue, we have glycosylation positive viruses. And when we grew these viruses on regular cell culture, there was no difference in replication. This is infectious virus production and time. But when we put these viruses in our nasal epithelial cell culture systems, you suddenly see a major difference in replication. What was surprising to us is that the virus that emerged, the glycosylation positive virus, was replicating less effectively than its parental virus. Counter to what we would have expected. We would have thought that that virus was a better virus at replicating because it outcompeted its parental virus. So when we looked at the neuraminidase activity of these viruses, it also surprised us that the glycosylation positive virus had a lower neuraminidase activity than its parent, shown here in red. So again, a little counterintuitive that less activity of the virus, of the enzyme, was associated with a more successful virus. We finally found a positive aspect of this mutation when we looked at the ability of antibodies to bind to and inhibit neuraminidase activity. And in this assay, we were able to show that it took much more antibody to inhibit the neuraminidase activity of the glycosylation positive virus when compared to the glycosylation negative virus. 16 of 20 individuals had reduced NAI titer antibodies against this NA-gly positive virus. So in this 2016-17 season, we really think that the NA protein underwent antigenic drift. Carbohydrate was able to block an antibody binding site that provided the virus with an antigenic advantage. But that advantage came at the cost of virus replication as well as neuraminidase activity. So in this case, escaping pre-existing population immunity was more important than viral fitness. Move to the next year. And in red here are the cases of influenza in the 2017-18 season. And you can see a massive difference in terms of the amount of cases here, with peaks also at the end of February and March, a large peak around the Christmas holidays, but as you can see, much higher levels of cases than in any year since the 2009 H1N1 pandemic. The viruses that circulated here were the same H3N2 viruses, but I'll get a little bit more detail about those clades. HA clades are very similar to what you hear about with COVID-19 and SARS-CoV-2 variants. They're simply mutations in the spike protein that allow us to differentiate different viruses from each other. With influenza in this year, the H3 viruses really consisted of 2 groups of viruses, a group called the 2A viruses, which are shown here, These have accumulated mutations to make them distinct from each other in the HA gene. There was also a virus called a 3A clade, which was a more ancestral virus that continued to circulate, particularly on the east coast of the United States. So when I say an H3N2 season, it really was a more complicated season than that because we had these 4 variants that were all detected and circulating. Now, when we look at the viruses from 1617 to 1718, we see a switch from the 2A1 to the 2A2 viruses as the dominant clade. But when we looked at the ability of those HA proteins to be recognized by vaccine responses, they were exactly the same. So that suggested that this switch was not because the virus could escape vaccine immunity any more easily. But when we sequence our viruses, we don't just sequence the HA protein, we sequence the whole genome. And what we found in this diagram is when we looked at viruses from 2016-17, color indicates the clade, and here are the different segments. Viruses from 2016-17 show the same color across the board. They were very homogeneous, whether they were the 2A group or the 3A group here. But viruses from 17-18, you see the different color patterns here. This suggests that the different segments from different parental viruses were part of their genomes here. And the 3A viruses are shown down here in this blue and green. To simplify that, we think that in 2017-18, the 2A2 viruses actually had 2 segments from their 2A2 parental virus and 6 segments from the dominant virus in that previous year, the A1 virus, representing a reassortment event. The 3A viruses had a very similar reassortment event. 3 segments were conserved from the parental, but 5 segments came again from that dominant virus in the 2A. So if you simply looked at the HA protein, you would think the same viruses were circulating in 2 years. But because we do whole genome sequencing, we were able to tell that other segments in the 17-18 viruses were very different from the parental viruses that were circulating in the prior year. What difference does that make? Well, with the 2A2 viruses, in purple is the reassortant virus. In green and orange are the 2 parental viruses. These are replication curves on our HNEX. You can see the purple reassortant virus replicates to much higher titers than either of its parental viruses, which gave its segments. Interestingly, that virus I first talked about is shown in green here. So reassortment allowed this virus to increase its replication to levels that are consistent with viruses that circulated in previous years. Interestingly, when our clinicians started to look at disease severity in this population, they sorted out a population of individuals that were suffering more severe disease from influenza in this year. And it turns out the majority of those individuals were infected with this reassortant 3C2A2 virus. And when compared to the non-reassorted virus, this reassorted 2A2 introduced more severe disease in infected individuals, correlating, of course, with our culture replication here. Now, with the 3A virus, it was a little more complicated. And here, the parental virus is in purple, the parental is in blue. The reassortant is in purple, the parental is in blue. And you can see that here, there was no replication advantage based on reassortment. The parental 3A and the reassorted 3A replicated the same. And there was no change in disease severity between people infected with the reassorted or the parental 3A virus. But I'll come back to that 245 NA glycosylation. The 3A virus had previously not had this 245 glycosylation site. Reassortment brought the glycosylated NA protein into this genetic background. And now we can see that the reassorted NA viruses were much less sensitive to antibodies that recognize the NA, whereas the parental virus was highly sensitive to those antibodies. So reassortment in this flu season happened in 2 different H3 viruses with 2 different results. In one set of viruses, it increased virus fitness, and in another set of viruses, it increased the NA antigenic drift, allowing more people to become susceptible to infection. So again, I'm going to close by simply pointing out that whole viral genome sequencing can provide important insights into the phenotypic changes that drive increased circulation and disease severity. Machine learning methods in our emergency department can help identify the small percentage of individuals who have progressed to severe disease, and we're working now to integrate our viral genomics in that to try to understand what viral determinants might be pushing changes in disease severity. And then artificial intelligence-driven modeling of nonspecific symptoms helps prepare medical and public health responses to surges in respiratory infections. Again, those last 2 points I didn't get a chance to talk about much more, but I'd be happy to talk about it more during our panel discussion later. And with that, I'll thank you very much for your attention.
So next speaker is Saskia Skubik. She is a group leader in Department of Biology, Faculty Science in Utrecht University, Netherlands. She has developed a state-of-art organoid-based co-culture system uniquely tailored to model intricate interactions between cell populations, exhibiting diverse cellular fitness. She's worked on innovative 3D organoid co-cultures to combine quantitative imaging and cell biology to answer how cell competition safeguards tissues. Saskia, please.
First of all, I would really like to thank the organizers for inviting me to be here in this beautiful city, talking about great science, and I'm really looking forward to spending some more time with all of you and learning about the great things that are being done here in Korea. So, as said in the kind introduction, my team is really interested in how cellular interactions contribute to tissue physiology. And we're specifically interested in a process that is, in origin, crucial for early development, named cell competition, and how this plays a role in adult tissues. So as I mentioned, we're interested in a process called cell competition, which means that within tissues, which are basically all a mosaic, one way or another, cells continuously compare themselves. And those that are stronger will identify those that are weaker and eliminate those. And this will result in a situation where only the fittest cells will remain in a tissue and survive. And as you can imagine, this is a super important mechanism for quality control. This starts during early development, but it remains to be of a major importance throughout adult life. It can also protect against disease, for example, through elimination of early transformed cells from epithelia, but also in the thymus it can protect against leukemia. However, with many of these processes that are important during early development, there's also a backside to this. And we and others have found that cell competition can in turn be used by tumors and hijacked and used in their own benefit. And this is specifically what my team is interested in. And when I talk about cells compare themselves and they eliminate each other, what do I mean? This is all driven by differences in cellular fitness. So again, it's fitness. In this time, it's not viruses, but we're talking about fitness of cells. And what this actually means, I would like to illustrate by two pieces of work that I contributed to as a postdoc in a lab of Eugenia Pedini at the University of Cambridge in the UK, where we studied cell competition processes in the Drosophila midgut. And what we found is that those cells that carry a minute mutation, which have a ribosome impairment, are eliminated by surrounding enterocytes. However, the same wild-type enterocytes themselves will be eliminated when they're facing tumor cells with an APC mutation. And this means that genetically identical cells can either be a winner or a loser. It's the environment that determines the outcome of the process. So in other words, it doesn't mean how strong, it doesn't make, it's not important how strong you are, it's important how strong your environment is, and this determines whether you will survive. So what we use in my lab is organoids models, and they have already been introduced, so I can skip over the large part, but we're specifically studying adult stem cell-derived intestinal organoids. And what we developed is a co-culture system that we can use to really study these cellular interactions. And the principle is very simple. You take a population of organoid cells that are of a certain cellular fitness. For example, the small intestine organoids that I show you here. You give them one particular color. You take another organoid population with a different cellular fitness, and you give them a different color. You break them into pieces, and you fuse them into hybrid organoids. And these we can then study by looking at pathway activation, long-term time-lapse microscopy, or by then sorting them and doing omics approaches to really study these cellular behaviors in time on a single level, but also on a population scale. So what does it mean? These are two examples of organoids that come from our assay. So on the left, again, is the pure wild-type intestine culture, where you can see that this happily expands, it divides, the cells not only proliferate, but also they differentiate. You can see the formation of these script-like structures, indicating that all the different cell types that are present in the normal intestine are also present in our cultures. However, when we mix them with, in this case, colorectal cancer cells, which carry the most common oncogenic mutations that are found in colorectal cancer, we see a very different behavior of these wild-type cells. They no longer form these script-like structures, instead they keep their single monolayer epithelium, they don't differentiate, and most importantly, they are gradually lost from these cultures over time. And this is particularly obvious when you look at the quantification in the graph on the right, where you can see that at the end of the experiment, these mixed wild-type populations actually are lower in number than they were at the beginning of the experiment. And this means that it's not just a passive process that occurs, but in fact it's an active elimination of these wild-type cells. So when we further looked into this, we indeed could see that these competing wild-type cells show higher levels of apoptosis, indicating that they're actively eliminated through induced cell death. And most importantly, when we treated these cultures with small molecule inhibitors that prevent apoptosis, we can protect these wild-type cells from being eliminated. And this, I think, is very important and very illustrative of how complex these interactions are. Because by protecting this wild-type population, the cancer population actually loses the capacity to overgrow and overcolonize this tissue. And this means that even though these cancer cells that intrinsically have a higher proliferation rate do not get the possibility to overgrow and colonize the tissue when the surrounding tissue is not permissive. So it's really a two-way communication that is important here. Of course, there's not just one population of cells in these organoids, there's also the cancer population. And interestingly, what we noticed is that when we measure the expansion rate of these cancer cells, even though, as I mentioned before, their intrinsic proliferation rate is higher than of the wild-type population, this is even further increased when they're in mixed conditions. So again, there's a two-way communication. On one side, these wild-type populations get a negative influence from the cancer population, which starts to eliminate them, and then these signals are then used by the cancer population to increase their growth. Because of time, I do not have the opportunity to go into the molecular details of everything that is driving these processes, but feel free to ask me during the Q&A or come and see me in the breaks, or check out one of our publications, which goes into more detail here. But just in short, to summarize this part of the talk, what I've shown you is that in primary colorectal cancer, there's a continuous competition between the two different cell populations. And on one hand, the wild-type population is being eliminated by the presence of these cancer cells, which in their turn use these signals to boost their growth. If we inhibit these competitive interactions through many different pathways, we gain two major effects. On one side, we can protect the wild-type tissue, so there's no organ failure in the long run, but also, more importantly, we take away this pro-proliferative advantage that these cancer cells get from these competitive interactions. Okay, I'll try to be quick because I would like to talk about another story that we've been working on in the lab, which is the further stages of cancer development. Because even though colorectal cancer is one of the most common cancers worldwide, primary colorectal cancer is not necessarily directly harmful for patients. What is most destructive is growth in secondary organs. And when we talk about metastasis for colorectal cancer, this is mostly in the liver. So what a PhD student in my lab decided to do is to test whether we could model these competitive interactions in liver metastasis as well. So what she first did is isolate organoids from the mouse liver, and she mixed them in a similar way with the same colorectal cancer organoids that we used in the primary intestinal cancer. And I must say that for the first experiments, we worked with liver progenitor cells, but I'll continue with a more physiological system in the last slide. So what do we see? First of all, when we focus on the cancer population, we see a very similar response. Again, the cancer population itself is more proliferative than the wild-type progenitor cells. However, by mixing them with these liver cells, they gain a competitive advantage. So somehow these cancer cells, it doesn't matter which cells you give them, as long as they're weaker, they gain a competitive advantage. What happens with the wild-type cells? Actually, something very different. You can immediately see from the graph in the middle that the expansion rate between the wild-type in pure conditions and in mixed conditions is very different. However, the slope is also very different to the competition effect we saw in primary intestinal cancer. And in this case, it flattens, but it doesn't decrease. And this immediately told us that these cells do experience competition. However, the effect is very different. And this is not caused by induction of cell death, because then you would expect this decrease. Indeed, when we add small molecule inhibitors to prevent apoptosis, it doesn't do a thing in this system. So something else is happening. And of course, when you see a graph like this, you can immediately envision that there's an effect on cell proliferation. So we used multiple assays to visualize this, but I think the most beautiful one is using time-lapse microscopy again. So what we used is the Fuji cell cycle reporter system that might be known for some people here, which is based on two fluorescent readouts, one for G1 phase of the cell cycle, which is shown in magenta, and one for the SG2M phase of the cell cycle, which is shown in yellow. So basically, when cells cycle through the cell cycle, they switch colors continuously. And this is exactly what we see in the left, where we visualize the cell cycle progression of pure wild-type progenitor cells. And they indeed switch between magenta and yellow continuously. However, in the mixed organoid on the right, we see a very different pattern. Initially, at the beginning of the time-lapse, you can see quite a few yellow cells, and they come from magenta and they keep on cycling. However, towards the end of this time-lapse video, you see a massive decrease in the number of yellow cells. And this is specifically clear when we look at the last frame of this time-lapse. There are barely any Venus-positive cells anymore, indicating that these cells have completely exited the cell cycle. And indeed, we can see these black holes, which there are still cells there, but they just completely lost the cell cycle reporter, which indicates that they're in a G0 state of the cell cycle. Of course, there's a very strong link between inhibition of cell cycle progression and differentiation. So that's the obvious next step we wanted to evaluate. And one of the ways we did this is by going back to stainings for different markers. And here I show you an example of progenitor marker SOX9, which is a transcription factor, which is very high in these pure wild-type liver progenitors. However, in these mixed conditions, we see a massive decrease in the staining of this marker, indicating that indeed, upon competition with metastatic colorectal cancer cells, liver progenitor cells are forced to stop dividing and then to differentiate. Well, unfortunately, the assays that we do here, we can push towards like 5 days maximum, but this is not enough time to fully differentiate liver progenitors. So that's why we chose to use an artificial or a different approach, where we first force these cells to differentiate using specific cocktails of media into hepatocyte-like cells, and then use these for competition assays. And when we did this, basically the assay went too well. Which in this case was very unfortunate, because what we saw is that at the last day of the experiment, on day 3, in the differentiated conditions, basically all organoids are only made of cancer cells. So these hepatocyte cells are so efficiently outcompeted that we basically couldn't trace them. And of course, this is very far away from physiology, because if you think about an organ containing metastasis, it will never be a tumor with a couple of liver cells. It's the opposite. So that's why the PhD student in my lab decided to explore these assays further and use her knowledge that she got from her previous experience. And this actually really resonates with the first talk of today, because indeed things need to flow. So we went back to suspension. We took them out of the matrigel, and instead of growing them in organoid-like structures, we grow them in microtissue-like structures, which have circulating medium as well. So yeah, it's clearly depicted here. You break them into pieces, but you really make sure that all the matrix is gone before you can do this. And what we get is these like liver microtissues that are 1 to 2 millimeters in size, so they're a lot bigger, and they really mimic the physiology of the liver a lot better. And when we started using these for our assays, we saw a different picture. So these are 2 examples. You can clearly see that the liver, pure liver, is viable and healthy. But when we add the colorectal cancer cells in these mixed conditions, we see that they really start to integrate. And more importantly, when we follow the behavior of these cells, we see that there is a loss of the wild-type volume in these mixed microtissues, indicating that, again, there are competitive interactions at play. And when we now start to follow the behavior of these cells, we see that there's a high level of apoptosis. And this specifically happens at the interface between cancer cells and liver cells, indicating that some kind of competitive force coming from the cancer population induces cell death of the surrounding liver cells, and this causes their elimination. And one thing that I would like to finish with is, again, that to show you that fitness is really relative and that these interactions really determine how well you can behave as a cell population. Because what my PhD student noticed is that when she tried to make these microtissues from pure cancer cells, they actually really wouldn't form. They're very unhealthy, they're very fragile in structure, and they really needed some kind of support. And this was given by these liver cells. So by adding a scaffold, these cancer cells could survive. So what we see here is actually a 2-phase competition strategy, where first these cancer cells use the hepatocytes and the matrix they make to form initial structure, and then they start to eliminate them and use the goodies that they release to eventually grow better. Okay, this was a sort of superficial overview of the models that we use. Of course, we really want to study the mechanisms that are at play here. And we have quite a bit of information on that already, so again, feel free to reach out. But just as a small summary and a bit of a tease, what I showed you today is that in both primary intestinal cancer and in metastatic intestinal cancer, there are competitive interactions at play. And on one side, we see that the wild-type intestine cells are actively eliminated through induction of cell death. While in the metastatic intestinal cancer, we first see induction of a proliferation arrest followed by differentiation, and then the cells can start to be eliminated. In both cases, the cancer population really benefits from these competitive interactions. On one side, they proliferate more, but also they use these structures as a scaffold for their expansion. And I just wanted to point out that this, of course, has a major impact on potential cancer treatment, because, as I said, even though these cancer cells have an intrinsic higher proliferation rate, they can only use this when their surrounding tissue allows this. So there's a great opportunity to find out which signals they actually need and to block them. And with that, I would like to thank the people involved, and especially Anna Grotenberg-Garcia, who is on the right here, who did a lot of the work, but also the rest of the team, and the funding, and all the colleagues and facilities that really helped us. Thank you.
Okay, I will introduce the next speaker, Ki-Hyun Lim, from University of Sydney, Australia. Dr. Lim has developed a number of research technology platforms, primarily using photopolymerizable hydrogel bioinks for 3D bioprinting, of functional tissues and also delivery of bioactive molecules to promote tissue regeneration. Dr. Lim's research focuses on adapting a class of polymers known as hydrogels as tissue engineering materials for a variety of applications. Today's topic is dynamic presentation of physical cues in tissue engineered constructs using biofabrication. Dr. Lim, please.
Well, thank you very much for having me here. It's really a great honor to join the talk concert, and of course looking forward to the BrainLink X Lab Day in Yosu for the next couple of days. Well, I guess what I really wanted to show today is really a short snapshot of what the group is now working on. So I'm a photopolymer chemist. A lot of my time has been looking into developing different materials as hydrogels to encapsulate cells, for cells to grow in them. The overall hypothesis is that the gels act as scaffold, and we're trying to mimic the native extracellular matrix to support the cells. And in the last couple of years, we've taken these gels and we've applied them as bioinks for a range of biofabrication, bioprinting technologies. And what I'm going to show today is how do we use biofabrication as a way to present physical cues, and physical cues in this talk will be physical architecture and also mechanical stiffness of these hydrogels and how cells would respond to them or how the tissue or the body will respond to them. Right, so one thing that I really hope that can get across as the take-home message is when you look at biofabrication, when you look at bioprinting, please be aware is that that means a range of technologies out there. So when someone says I've bioprinted a construct, you know, the go-to question should be what kind of technology they're using to print it. That's droplet printing. In our lab, we work on bioassembly quite a lot, lithography, extrusion, and they all have different pros and cons. So in this case, bioassembly, you can combine thermoplastics, you can inject different cells-laden materials, so you can imagine that the blue is one kind of cell, the red is one kind of cell, and start structuring them and layering them to try to mimic the native tissue architecture. Lithography is more using light-based cross-linking, so that's kind of my bread and butter. We develop hydrogels that can be responsive to light in terms of the cross-linking, and you can see getting different spatial resolution here. So each of these are different voxels. Each of this is 25 microns, so you can start pattern hydrogels in 25 microns. And extrusion is more the one most widely known. I think most labs have them if they work in bioprinting, where you can extrude hydrogels in different fibers and you can start stacking them again. So each of them have different advantages and disadvantages. What I'm going to start today is really major looking at extrusion, where when I started getting into this field, and this is about 10 years ago now, 2013, there was a seminal review paper from Professor Jos Mauda's group from UMC Utrecht. And at the time, we were really looking at how do we control shape fidelity. So the whole point of doing 3D bioprinting is that you're able to use the computer to design a model, and you're able to then print the model accurately, right? So if you print a construct that doesn't have any shape or doesn't maintain the shape, then it's kind of defeats the whole purpose of going through a very lengthy and laborious process of trying to print the construct. So at that time, a lot of people were looking at how do you get this shape fidelity, and the easiest way to print the construct with really good shape is to have a hydrogel that has very high stiffness. So if you have a high amount of polymer content in it, you can print it really well. But we do know once you actually put cells in them for the cells to be able to remodel the matrix and to grow and elongate, they don't like a very stiff environment. They like an environment that is much softer so that they can remodel. So this is just really showing in a stiff gel, if you encapsulate cells majority of the time, they just stay around it. If you give them enough space for them to grow in terms of just endothelial cells, they can start forming these microcapillaries within them. But when you look at the different shape fidelity, this is where you want to have a hydrogel that you can start stacking and you can hold and fiddle around. If you don't have the shape fidelity, then you just get this kind of blobby structures that you can't do anything with them. So the trend in the field at the time was really looking at the novel strategies. How can we find technologies that you can print good shape and really permissive environment for cells to actually to grow? What we did is completely opposite. Instead of these novel strategies, we just went Why don't we just go indirect printing? Do we really have to directly print these materials, or can we use a sacrificial approach to indirectly print them? So I'll show you a little bit of what I mean about sacrificial. So there are materials out there that already are known to have really good rheological profile. So pluronic, for example, is really well known. What you can do is print them, you can stack them, you can then deposit or extrude a secondary material, you can cross-link the material, and you can wash your sacrificial template out. So by doing that, you have very good control over your sacrificial template, and the material then would then solidify and stabilize with that physical architectures that you are actually interested in. So we started with silk. The fibrous part of silk, the fibroin component of silk, is a very, very well-known biomaterial, very long-lasting in the field. This is a collaboration with Professor Jelena Renek-Kovacina at the University of New South Wales. And we were interested in using silk as a way to deliver biomolecules, deliver cells, and can we make them into hydrogels in this case. We've recently also just published this review. If anyone is interested, you can have a look at the way that we're controlling cross-linking in silk. So the idea is you print a pluronic solution, a pluronic scaffold in this case. Within this now second nozzle, you have your silk solution. Within them, you can have cells. You can then cross-link this whole material, and you can wash your pluronic out. So because pluronic has this really good rheological profile, you can control the design. So you can do designs of just simple lattice structures, 0, 90 degrees. You can also do more complicated design, like 0, 45, 135, 90 in each of the stacking as you go in different layers. And what that really gives you, when you wash your sacrificial template out, this is a silk hydrogel. So this is uniform fibers, uniform channels within them, or you can also have thin fibers, big channels, small channels, all in one construct. So if we're thinking about controlling physical architecture, or even porosity in this case, you can do that with sacrificial templating. And really, because the sacrificial material that we're printing is very well known to have really good rheological profile in this case. So what we did is something like this. You can form a gradient structure. So this is just a silk construct where you can have different resolutions of fibers. It goes from 40 microns, 75 microns, 120 microns, all in one construct. And you can also space them apart, 100 to about 400 microns. And we know that sacrificial printing is cytocompatible, so you can put cells in them. This is just human articular chondrocytes that we put in them. And this is after 28 days of culture, so the cells stay alive. And we had shown a lot of tissue matrix deposition, and also GAG per DNA study in this paper, which you can have a look later as well. Now, you might be interested to know why are we fixated on tissue architecture. And this is some work that has been published in the field some time ago by Buddy Ratner, looking at porosity, how porosity affects how tissue is going to respond to it. What we were interested in this case in silk is that silk is known to be a bioinert material. And it is known that if you implant silk scaffold, or a hydrogel, you normally get fibrous encapsulation. So the body doesn't quite want to do anything to it, it just kind of encapsulated, right? So in this case, if we impart some sort of architecture into these silk hydrogels, you can have silk that has no channel. This is kind of work that was being done by David Kaplan's Good and Tufts, where they normally just punch needles into silk hydrogels to get parallel channel to kind of direct cells to grow in one direction. But with sacrificial templating, what can we do here is to have interconnected channel. So now you have a silk construct with completely open pores, and they're all interconnected using this method that I've just showed earlier. What we do know that if you have interconnected channel, you have a higher surface area to volume ratio. So if cells are going to interact with it, there's more surfaces for cells to interact. So when you sit cells on them, you can see much higher proliferation going on with them on this interconnected silk scaffolds with interconnected channels within them. And when we implant this subcutaneously, just in the red in this case, you can kind of see without any channels, you get fibrotic encapsulation. With the parallel channel, you do get tissue infiltration into the hollow channels that you punch into them, but you still get a fibrotic capsule around as well. But in the interconnected channel, we now no longer see that fibrotic encapsulation. So this is really showing that not just material is driving how the tissue is responding to it, but actually material and architecture is driving how the host tissue responds to these materials. And when we look at tissue infiltration, because you have more surface area, you get more cells growing into them, you get more tissues deposited in them. So this is two of the papers that summarize this work on here. Now, with another collaborator at Harvard Medical School, at Shrike Zhang, we started looking into, can we actually compare and control how we dissolve this sacrificial material. So when I showed about pluronics, this is what we get, right? So you print a sacrificial template, you put a secondary material, instantly all of these channels are going to open the moment you actually put them into cell culture media. But if we control the chemistry, we should be able to block this channel. So initially, the channels will be blocked. And depending on the chemistry that we use, we should be able to delay when these channels are going to slowly open. So would the dynamic presentation of these physical cues actually also influence how cells are going to behave? So we were just doing extrusion printing in this case. We're using gelatin. My lab uses gelatin quite a lot, mainly because it's cheap. It's also easily to modify using different chemistries that we have. And in this case, we know if you cool the gelatin down, it becomes a jelly. If you heat it up, it's a solution. For those who bake, for those who cook with gelatin, this is kind of common knowledge. And in this case, we heat the nozzle here. So this is heated up to about 30 degrees. This base plate is cooled down to 4 degrees. So the moment you extrude your fiber and it touches your base plate, it solidifies and you can start layering them, right? And using a specific light-based cross-linking method, we are also forcing or causing dityrosine cross-linking between the gelatin polymer chains. So we are introducing some covalent cross-links within them to kind of control its dissolution. So now we have a material that when it's cold, it is a jelly. When it heat it up, it remains as a jelly, but not for long. So with time, this dissolution now just takes much longer. So if you look at printing gelatin, it prints really well. So a 10% gelatin also has really good rheological profile, right? You can extrude them, you can stack them. This is just showing different architecture, different fiber diameter from 0.4 to 0.8, all in one construct. And this is what I meant about delaying the dissolution. So by just having additional cross-links, your gelatin now doesn't degrade or dissolve instantly. Instead, it can stay all of the way up to 14 days and only then dissolve. This is kind of important. So comparing to what we showed at the earlier stage with pluronics, where you get instant dissolution or opening of these channels, in this case, after 4 hours, these channels are still blocked. And you can control this, so you can have some scaffolds that only at day 3, the channels will open, or scaffolds where the channels will only open at day 10. So you can now control when you want these physical architectures to be introduced to the cells that are surrounded in the bulk. Okay? What this also shows us in terms of using them to microfluidic devices, for example, that you can also compartmentalize the flow, and you can have only one part of the flow that is perfusible at day 3, and then both of them perfusible at day 10, by just controlling the chemistry that we're using in this sacrificial printing. And this is also important from a tissue response perspective, where my lab does a lot of stuff on vascularization. So if you kind of mix your mesenchymal stem cells and your human endothelial, umbilical vein endothelial cells in this case, what we actually see is in a scenario that all of this channel is completely open versus a scenario that you block the channel for a while and then only open it and perfuse it, you can see much enhanced vascularization. So your average vessel length, your junction density is significantly much higher, and we do think that this is because when you block all of these, the cells are in a hypoxic environment, and what do endothelial cells do in a hypoxic environment is, of course, they try to form vessels. And then when you then turn it into a normoxic environment, they then stabilize. So by just controlling this microenvironment, we're able to see this much elevated response in terms of the vascularization growth in these vessels and these constructs. And we did prove that. So this is just, this is just showing the quantification of the cells in a hypoxic state. So in a delayed dissolution, you go from high amount of hypoxic cells turning into normoxic, instant dissolution, you have it the other way around. So what we have now is being able to control microenvironmental cues by just controlling when we are going to introduce these pores or these physical architectures into this construct. This is kind of a platform, so we also have published this work where the sacrificial or the delayed dissolution sacrificial templates can be used to a range of materials. It can be with polyvinyl alcohol materials, for example, different cross-linking, light, redox-based, chemical initiated. So this is kind of an interesting project that we're having ongoing in the lab. Very lastly, I think I spoke about mechanical stiffness as well, and just looping this back into the silk story. We do know that silk proteins undergo assembly in when just incubated in media in the presence of salt. And we do know that this silk goes from a random coil to a bitter sheet transformation. And in this case, what we really wanted to do is combine this silk with some of these adipose-derived decellularized extracellular matrices that we obtained with a collaboration with Professor Gina Zhang's group as postdoc here in Pohang. And using this, we are able to make hydrogels that would slowly increase in stiffness over time. And this really goes back to the whole story that another different research arm that I have in the group where we're looking at cancer mechanics and how disruption in mechanical homeostasis is kind of a hallmark of cancer progression. So how do we control the change in the dynamic change in this mechanical stiffness in terms of modeling cancer growth? And this is just showing with cells, you also get this dynamic stiffening in these gels. And I think this is kind of interesting because all of these different formulations have no stiffening effect. Only this one with the yellow arrow have a stiffening effect. And you can see that the tumoroid that is being grown in here, so this is MCF7, breast cancer cells, and you can see that you kind of have this layer of epithelial cells outside, which is luminized. And what we think is by just changing the mechanics, we're able to capture this progression. So now we're really able to capture this middle stage of this tumoroid growth. And when we look at epithelial mesenchymal transition, we're also able to see in the stiffening part where only the outer layer remains is epithelial phenotype. You have your E-cadherin, and in the middle, the cells now start secreting fibronectin, so they're turning mesenchymal. So we're able to capture cancer progression by just controlling that change in mechanical stiffness. And again, this is work that we've published this year. Right, so just to quickly summary, I think what I wanted to show today is that there are a number of different bioprinting technologies out there. So when you look at biofabrication or bioprinting, you know, the go-to question should be what technology are we talking about? What I've showed mostly is in extrusion printing, where you can use extrusion integrated with sacrificial templating to start introducing different physical architectures into tissue-engineered construct. And using that, you can also control the time when you want these physical architectures to be actually introduced to cells, and we showed that by doing that, we can enhance the formation of vessel cells. We also showed chondrogenesis, adipogenesis, and osteogenesis as part of the paper, which I don't have time to show. And lastly, by just controlling mechanics, we can also look at using that to model cell progression. In this case, what we did was cancer modeling. Right, so I have to thank my group. A lot of this work was done also when I was still in New Zealand in the collaboration of Professor Tim Woodfield and a lot of my different collaborators. So thank you very much.
Okay, I will introduce the fifth speaker, Professor Young Charles Zhang. He's from Emory School of Medicine. The multidisciplinary approach to study muscle cell biology and develops bioactive stem cell delivery vehicles for use in regenerative medicine. Dr. Zhang's lab studies both basic aspects of stem cell biology, especially systemic metabolic regulations of stem cell leaches, as well as more translational aspects of muscle stem cell and mesenchymal stem cell for use in therapeutic approaches for musculoskeletal aging, neuromuscular disease, and traumatic injuries. Today he will talk on the integrative stem cell engineering to rejuvenate aging skeletal muscle. Dr. Zhang, please.
Okay, I also like to thank the organizer for the invitation, and I'm really looking forward to the discussion later on. In a nutshell, my lab studies aging, especially skeletal muscle, and my first name's Young, so it's when you study aging, having a name like this actually helps. So I'll start with a little bit of introduction on aging. I'm sure everyone in the audience knows that our demographic population is shifting towards aging. And this is data from US, but actually in Korea it's much faster. So if you calculate this in math, actually our life expectancy is increasing 6.5 hours a day. So chances of becoming a centenarian is less than 1%, but I guess the students over here has more highly chances of becoming a centenarian. And As I mentioned, so our demographic is changing. Again, this is data from US. Within the next 10 years, there's going to be more people over 65 than people under 18. So if you live long and live healthy, it's not going to be a problem. But actually, living long will lead to age-related diseases, and there's going to be a lot of age-related disease healthcare costs. So my lab is interested in skeletal muscle aging, and it's termed sarcopenia. It's a loss of muscle mass and function independent of aging. As you can see from the image from MRI of your thigh, if you compare a 20-year-old to a 70-year-old, there's a significant decrease in muscle mass. And when you lose muscle mass, it's replaced by subcutaneous fat. So there's a lot of metabolic dysfunction that occurs with the loss of muscle mass. But when you also think about muscle, in humans, there's over 600 different muscle types. And in our body, there's muscles everywhere. And when we think about muscle, we only think about contraction. But actually, there's a lot of interaction from muscle to other organs. And just like hormones, muscle release factors like myokines. And these myokines will go through the circulation and act on other parts of the body as well. But also, the second form of communication is actually electricity. So muscles contract due to the innervation from the peripheral nerve. And in this way, we could also control the muscle to central nervous system as well. And this is very important for maintaining a health span. So this is data from Professor So, which he'll be here tomorrow. So the more muscle mass you maintain at the later stage of your life, actually you could control. There's chances of decreasing age-related diseases, and actually death could be decreased by maintaining a muscle mass. So why we age and why muscles age, there are more than 200 different hypotheses or theories of aging, but if you combine all of those, it boils down to this 12 areas. And within this 12 area, muscle is a similar way, and my lab is particularly interested in how the stem cell dysfunction would age. And as Professor Luke Lee mentioned, mitochondrial dysfunction also lead to an aging process as well. But due to interest in time, today I'm just going to talk about heterochronic parabiosis, where we surgically attach young and old mice. So instead of mixing the cells, we actually mix the whole animals. So to give you a little introduction on the muscle stem cell, so I'm sure everyone saw this type of images in the textbook. Muscles are made of fiber bundles. And within this muscle, if you look at the single muscle fiber, one of the unique things about muscle is that it's multinucleated and it's post-mitotic. So they don't divide, and the stem cell expressed by PAX7, the pink cells here, actually add more nuclei as we go on. And if there is an injury to it, actually this cell type plays an important role. They actually go to normal physiological condition, they stay in quiescence, but when there's injury or there's a cue, they enter the cell cycle and go through the proliferation and differentiation phase and makes a post-mitotic myotube, and this myotube actually fuse to the site of the injury. So for injury repair, these muscle types are very important. But with age, we use mouse model, C57 black 6 mice, the mean lifespan of about 30 and maximum about 40. If you compare the 2-month-old to 40-month-old, there's about 50% decrease in total number of muscle stem cell. And not only that, whatever that's left at the 24-month-old, they can't form a new muscle fiber. It's like a double whammy. So you lose muscle stem cells, and these muscle stem cells do not form new muscle fibers. So as a result, if you injure a muscle, so you're looking at a cross-section of the muscle, and regenerating muscle fiber has a centralized nuclei. So all of the young muscles, if you injure them, they will repair. But if you do the same injury to the 24-month-old mice, they have a hard time repairing, or they're very slow to regenerate. And so this is a common feature of aging tissue. Not only muscle, other tissue will have the same problem. So in order to get around this problem, one of the ways to rejuvenate aging tissue is called the heterochronic parabiosis, where we take the young mouse and the old mice and surgically attach so they share the circulation. And this type of surgery has been going on for more than a century, but we still don't know exactly what the rejuvenation effect of this surgical intervention. But if you compare these images, as I mentioned in the previous slide, young muscles, if you just focus on the bottom panel, the pink fibers are regenerating fibers. And if you pair the young animals together, the muscles will regenerate. But if you pair the old animals together, they don't regenerate. But if you pair the young and old again, they restore the regenerative capacity by simply exposing to the young systemic environment. So not only the muscle regeneration, other features of aging, such as DNA damage and myogenesis or the neuromuscular junction degeneration, have been shown to rejuvenate by simply exposing to a young systemic environment. So the million-dollar question is, what is that factor? And it's not just in the muscle. Other organs, simply if they expose it to a young systemic environment, they rejuvenate. So the million-dollar question is, what is this useful factor? So a lot of the research has been done. One of the classic study that I showed in the previous slide was done by my postdoctoral advisor, Amy Wagers and Arena Comboy. And when Amy started a lab at Harvard, she collaborated with everyone nearby in trying to find this factor that rejuvenates. And there are a lot of factors that actually rejuvenates, but there's also factors that could accelerate aging as well. So there's progenetic and antigenetic factors. But main problem with the heterochronic parabiosis is that it's really difficult to get therapeutic intervention out of this. And obviously, we can't do human parabiosis. So there's got to be a way to get around this problem. And a lot of people have done blood transfusion, mainly from Stanford, Tony Weiss-Corey. They did a lot of blood transfusion of the young and old. And to a certain degree, you could get a rejuvenation effect with the blood transfusion. But the main difference between transfusion and heterochronic parabiosis is that when you have two animals together, you're not only sharing blood, you're also sharing organs together. So there's a lot of hormones and other factors, especially the blood from the young bone marrow actually competes with the old bone marrow, and actually young always wins versus the old. So that's the main difference between blood transfusion and the parabiosis. To get around this problem, Professor Luke Lee gave an elegant introduction on the different 3D microphysiological systems. So we wanted to use this to mimic heterochronic parabiosis. So simply, we wanted to have a simple system. And in our case, our organ-on-a-chip system mimics the microenvironment of the muscle stem cells. So it looks a little bit complicated, but it's a double-layered channel. So upper channel has an endothelial cell layer. On the top, there's a circulation of the serum. And on the bottom, there's a side channel and a main channel. Main channel has a muscle system. So on the bottom, you have a myotube that mimics the myofibers. And on the top, you have a quiescent muscle stem cell. And the extracellular matrix is mimicked by the hydrogel. And on the side channel, we want to mimic the motor unit by putting the motor neuron. So we wanted to use this system to mimic and see if there's a serum effect and whether we can mimic heterochronic parabiosis. And one of the accelerated aging model is when you knock out a gene, SOD1, you have acceleration of the age-related muscle loss. And this loss of muscle mass is actually due to the increase in oxidative stress. And there's high oxidative stress in this knockout animal. So when you add when you compare the young serum and the old serum and SOD1 knockout serum, as you can see, the myogenic property significantly decreases in the SOD1 knockout mice. So it gives us a proof of principle to use this model to move on. So we took this system, and initially we used the microfluidic device, the same system with the upper channel and the lower channel, and we got the cells from the young animal, got the cells from the old animal, and compare a separate channel, and only the mixture of the blood from the young to old, old to young, or we mix the two systems together so we can mimic the heterochronic parabiosis. And using the system, we were able to mimic a lot of the things that happens in the in vivo. So we were able to see that proliferation, which decreases with age, and when we do the heterochronic parabiosis or the blood transfusion, was rejuvenated, and also the myogenic differentiation was significantly upregulated in the heterochronic parabiosis as well. And we also are able to find some of the factors that are responsible for this rejuvenation effect. And one of the factors that were actually VEGF, but this was just a proof of principle. I mean, we were only looking at certain factors. Obviously, there are a lot of factors. Well, so we went on and redesigned our chip, and because initial design was a microfluidics device, it was very difficult to do proteomics. So we made a different chip with the same system, and using a dedicated imaging system, we could combine the live cell imaging along with the proteomics approach, and we could do parabiosis with transgenic mice. Methionine tRNA synthesized mutant mice. So the methionine is a start codon. So if you mutate, if you give a point mutation of this gene, the newly synthesized protein is identified by the methionine ortholog azidonucleosin. So by tissue-specific matter, cell-specific matter, and time-specific matter, we could identify newly synthesized proteins this way that might play a role in the rejuvenation. And so we did the proteomics. We don't have a full picture yet, but a lot of the proteins that are remodeling the extracellular matrix were responsible for rejuvenating aging muscle system as well. And we're also interested in the systemic factor that rejuvenates the mitochondrial function, and we're continuing to working on this, but one of the things that we found is that short-chain fatty acid that's in the circulation is actually causing the mitochondrial biogenesis of the cells. So we're using intestinal organoid that we heard from the previous speaker, and we combined it with the liver and the muscle to really identify at what point, what factors are actually rejuvenating the muscle. So we're continuing to working on this and trying to get more data. So hopefully we could have a better picture of this. And lastly, one of the other ways to use the 3D microphysiological system is comparative physiology. And I don't know if you guys know naked mole rat. So I think there's a colony in Korea as well. So one is the unique thing about this animal is that normally the mice will live for 3 years, but these guys could live for 30 years. And one of the unique things is that they are resistant to cancer. So if you inject cancer cells, they will just resist, they will just kill the cancer cells. So by using this 3D microphysiological system, we could utilize the anti-cancer resistance of these animals. So we simply took the serum from the mouse and serum from the old, and kind of mimic the heterochronic parabiosis in the spiroid culture. And we were able to see that just simply giving the serum from the naked mole rat, the cancer growth was significantly was reduced. So we're continuing working on this. But more importantly, by using this in vitro system, we could mimic human parabiosis, and by combining single-cell sequencing and other proteomics approach, we could actually understand the rejuvenation effect of heterochronic parabiosis and hopefully in humans as well. So lastly, most important slide. So this in vitro parabiosis system was done by talented grad student Jungmin Choi. Now she's at Altos Lab. I don't know if you know, Altos Lab is the Amazon-backed up aging company, and she's doing a lot of rejuvenation work as well, and also other collaborators and funding sources. Thank you.
Next speaker is Xian Lee from School of Medicine, Stanford University. Dr. Lee is working at the Stanford Cardiovascular Institute to understand the role of endothelial cells in congenital heart disease using patient iPS cells. He's working on the identifying endocrine factors to control cardiomyocyte proliferation and maturation. Today's topic is unraveling the cellular and molecular mechanisms of non-compaction cardiomyopathy through single-cell RNA sequencing. Dr. Lee, please.
Hello, everyone. Yeah. First of all, I'd like to thank for inviting me here, and also honored to present my previous work and current work. So I got this title, and I'm actually the developmental biologist. So I always start with these images. You can see the different shape of embryo. So probably you saw the many cell or mouse, but I've been studying this embryo development for over 15 years. So in my PhD, I study endodome development, especially how the first liver cell or pancreas cell comes in the middle of this area. The second part, in my postdoc period in the Stanford biology, I study where is the first coronary vessel come in during the heart development. And currently, I'm the instructor in the Stanford CVI, and also I'm using the iPS drive, the patient iPS drive cell for studying congenital heart disease. And I just said even though I'm the development biologist, I'd like to understand the human disease, specifically congenital heart disease. Especially two types of the disease. One is LVNC, the cardiac disease where the patient has a very thin myocardial layer, and the other congenital heart disease is the Down syndrome patient who has the congenital heart disease. They have some defect in the septal period, which I'm going to introduce later. But my main focus is the more endothelial cell oriented topics. So I got trained under the Dr. Christy Redhorse, who is currently HHMI, and our main goal or also the cell type we are interested in is endothelial cells, specifically the origin of endothelial cell. There are multiple progenitor cell which can differentiate the functional endothelial cell, which is later undergo angiogenesis and arteriogenesis. Why these questions are important? Because the endothelial cell, the cardiovascular, the biology or this information is essential for understand the cardiac organogenesis, and to understand the disease, and also to know the recipe of the stem cell differentiation, and also the heart regeneration. So I always ask, where is the first coronary vessel endothelial cell during cardiac development, like a backside or front, because your heart or our heart is round, but we don't know where the first blood cell they come into the inside the muscle. So using the mouse genetics, previous researcher, including the Christy Redhorse, we found there are two different areas and two different endothelial cell type contribute to the coronary vessel endothelial cell. First is sinus venosus, is one of the progenitor cell located backside. That progenitor cells contribute to the backside. You can see the confocal microscope images. So this is back, more dorsal part. The other part is endocardial cell, which is located inside. They also contribute to more ventral side one. So in later stages, these two endothelial cell, which oriented two different progenitor cell, they merge together and they contribute to all the coronary vessel endothelial cell further artery. So my question of my project was what is the role of coronary vessel during the development? So when I studied this topic, I realized there were a big difference, especially the myocardial compaction thickness difference between these the developing period. As you see, from embryonic day 9.5 and 12.5, you don't see much difference in compaction or the outer layer difference. But after 12.5, you see the big difference. So my hypothesis is a coronary vessel endothelial cell may be an important role in the myocardial growth. You can see also here, there are no difference, but after like 12.5, where coronary vessel contribute. So this is overall my hypothesis. So as I mentioned, there are two different progenitor cells may contribute to the muscular, the endothelial cell. That may be the important for this thickness. However, to answer this one, I need model. So I generated new the mouse model. So I knock out chromatin modeler INO80, which is the chromatin modeler, but that gene has not been studied well. But because the INO80 knockout mouse died before like a implantation, so we generate the 3 different knockout mouse lines. So we knock out INO80 in the muscle, cardiomyocyte, and fibroblast, and endothelial cell. But interestingly, we didn't see any difference when you knock out this gene into the 2 different cell types, except endothelial cell. When you knock out endothelial cell, we see this cardiac structure is totally disrupted, and as I mentioned, we knock out this endothelial cell in the endothelial cell. That causes this non-compaction defect. But I emphasize, I knock out this gene in the endothelial cell, but the phenotype occurs in the muscle side. So previously, most cardiologists or many the people believe the LVNC is which is cardiomyopathy. So people believe this defect caused by mutation of cardiomyocyte. But I prove that this LVNC, this phenotype also occur solely the defect in the endothelial cell. So main part is as you see, when you section this heart, there are 2 parts. One inside part, trabecular cardiomyocyte, one of the cardiomyocyte which is elongated. The other part is a compact cardiomyocyte, which is located outer side, actually main side of the expansion. But when you knock out INO80 gene in the endothelial cell, compact myocardium proliferation was significantly reduced. And also, actually, this is one of the main contribution to the science of what I did. So to answer my question, people always ask, oh, you knock out the gene, but if you lose endothelial cell or something happen in the endothelial cell, probably that's because you don't have the proper endothelial cell with some defect in the flow. So flow may be involved, so you cannot answer your question properly. But I developed the embryonic mouse embryonic heart explant culture. So basically, I took out the heart at 12.5, and I can culture for 5 days, where we didn't the heart was not affected by the change of flow. So as you see, in the control side, the red is the cardiomyocyte, and this blue is the endothelial cell. When I culture, the cardiomyocyte and endothelial cell develop together. However, this is the mutant explant culture. There are only defects in endothelial cell, but we see the normal cardiomyocyte also defective by this one. So actually, I have used multiple mouse line and multiple experimental design. But main question then, what is the factor? It seems that I claimed there are some angiocrine factors, which is factors from endothelial cell. Can I identify that endocrine factor? So I thought the single-cell RNA sequencing analysis can identify the factors. Because especially this mouse heart is very small, so single-cell resolution can answer this question. So I ran these two mouse lines with the single-cell RNA sequencing with this hypothesis. So there may be some secreting factor that may promote cardiomyocyte proliferation, and maybe the other some factor that can cause reduced proliferation and induce maturation. So I designed with some validation experiment with the embryonic heart explant and human iPS cell-derived cardiomyocyte. So this is the single cell, the T-gene map after I ran the 10x single cell RNA sequencing. So there were like 9 different cell types, which I could identify. We could see including the fibroblast and endothelial cell and cardiomyocyte. And I subcluster only the endothelial cell, and that endothelial cell include the endocardial cell and coronary endothelial cell, and also one specific cell type which is highly upregulated in the INO80 mutant line. So using the histology method, I see some specific endothelial cells after we knock out INO80 that's overproliferative, and they have some issue in here, which validate in the histology. And also I try to find some factor which can promote cardiomyocyte proliferation. So I list up all the differential expression gene list, and I tested the top list. So I found one, the secreting factor, which is collagen 58-1, which is located, which is expressed in coronary vessel endothelial cells specifically, as you see in here, in the wild type. But this molecule is significantly reduced in the mutant coronary vessel endothelial cell, as you see in different graph. And to confirm whether this protein is this gene is essential or the change or promote the cardiomyocyte, I use the explant culture system. So when I treat the collagen 58-1 protein, I was able to see the cardiomyocyte has more proliferation, and the adding collagen 58-1 into the INO80 knockout, I could see the rescue experiment. And also I specifically inhibit the endothelial cell. So as you see the endothelial cell removed, there are like a similar LVNC phenotype, but the adding collagen 58-1 the partially rescued this phenotype, and also the collagen 51 add to the LVNC human iPS patient iPS cell derived cardiomyocyte tested, and it also partially rescued the mutant one as well. So this is overall. So actually, I didn't show all the mouse line, but when I knock out the INO80, one of the important gene for the endothelial cell, I was able to recapitulate the LVNC human congenital heart disease model, and also single-cell RNA sequencing identify some gene, including collagen 51, for the promoting cardiomyocyte proliferation. And also I didn't show the molecule which also involved in the maturation. That is one part, and currently I'm working on the other congenital heart disease model, which is the Down syndrome congenital heart disease model. Like Down syndrome patient, 50% of Down syndrome patient undergo this ventricle septal defect. So, however, there is no way to study this one because this developmental the phenotype or the disease. Luckily, the Stanford Biobank, especially my mentor, Joseph Wu, established the Stanford Biobank, which has 3,500 the patient IPS line. So I was able to generate cardiac organoid, and using multiomics and spatial transformics, I'm continuing this one, and to show the some kind of the proof why I think the endothelial cell is also important for septal, which is this part is the. So I revisit the endothelial tied to Cre-ionoid flux line. You can see the also when the endothelial cell is some disrupted, the septal part is also affected. So as you see that during development, not only this ventricle part, this septum part also the grow, but this part is also affected by the endothelial cell. These are all the references I've been chasing, and other evidence. I knock out VEGF receptor 2 in different Cre line, and I also generated this the ventricle septal defect phenotype. And this is kind of the working proposal. This coronary, one of the endothelial cell, which is endocardial cells, are the crucial for the normal septal development. As I mentioned, see, we are generating the mini heart, like a cardiac organoid. So using like a 20 and 20/20 patient lines. And also, this is the example. We are able to generate the cardiomyocyte from all the patient line, and also the endothelial cell, especially focused on vascularize the cardiac organoid. And this is different media to show the normal media with the cardiac organoid without the media essential for endothelial cell that affect also the cardiomyocyte, our cardiac organoid expansion. So for the summary, I show the coronary vessel endothelial cell is important for the two different parts. One is ventricle part and septal development, which is also important for the congenital heart disease. And I established the embryonic heart explant culture and iPS cardiac organoid to study this congenital heart disease. Single-cell RNA sequencing also important tool to understand this mechanism, and especially also these days spatial transformics technology help we understand specifically the region phenotype. So I was able to revisit the published data set. We reanalyzed this set. We see the endothelial cell also kind of important for some LVNC or the septal defect one. So I'd like to thank my previous boss, Chris Redors, and current PI, Joseph Wu. Yeah. Thank you so much.
I introduce next speaker, Michael Zhou, Dr. Michael Zhou. He's from Iowa State University College of Penn Medicine. He's working on novel vaccine strategies to elicit broadly neutralizing antibody against the fusion peptide of HIV GP41. He's working on the structure functional of viral envelope glycoproteins such as virus host inactions, molecular mechanism of virus entry, structure-based immunogen design, and vector and platform development for antigen delivery. Today, he'll talk on the novel vaccine strategy to elicit broadly neutralizing antibody against the fusion peptide HIV GP41. Okay, Dr. Zhou, please.
Okay, good afternoon, everyone. I also would like to thank the organizers for the kind invitation. I got here in Korea last night, so I'm still a little jet-lagged, and it's 2 a.m. in the morning, where I came from. So apologize in advance if I sound a little dozy. Okay, so today I'd like to talk about novel vaccine strategies to elicit broadly neutralizing antibodies against the fusion peptide of HIV-1. But I woke up 4 in the morning today, and when I was looking at this title, I said, maybe this is a little too optimistic title. So the actual title should be like this, instead of broadly neutralizing antibodies, just neutralizing antibodies. Okay, I have to disclose that there is a financial conflict of interest in that I have an equity interest in NeoVaccine Incorporation, which is a company that I founded, and I currently serve as the CEO and president. So just to give you a little quick introduction, it's been now 40 years since the AIDS pandemic started, and unfortunately there's still no vaccine. We all know that we had a SARS-CoV-2 pandemic, and we were able to come up with a vaccine within less than a year. But for HIV-1, we still don't have a vaccine even after 40 years. All phase 3 AIDS clinical trials that were conducted during the past 25 years have failed to show significant efficacy. And in my humble opinion, all of those vaccine trials have failed because none of those vaccines were designed to elicit broadly neutralizing antibodies. During the past 15 years or so, dozens of broadly neutralizing antibodies have been isolated from HIV-1 infected human patients. So that tells us that it is possible to elicit broadly neutralizing antibodies. We just have not been able to figure out how to do that in a vaccine setting. So the induction of high titers of potent broadly neutralizing antibody is the most important goal towards developing a vaccine against HIV-1. And towards that goal, we have been working on designing novel immunogens, developing innovative vaccine strategies, and exploring non-conventional vaccine delivery platforms. And today I'm going to talk about ongoing efforts to elicit broadly neutralizing antibodies against the fusion peptide of HIV-1 GP120. And I emphasize ongoing, which means that we haven't figured out yet, but we're working on it. So the fusion peptide is located, where's the? Okay, so fusion peptide is located at the very N-terminal end of GP41 subunit of envelope glycoprotein. And what I'm showing here on the left-hand side is that trimer of GP120, GP41 heterodimeric structure. And the GP120 is shown in gold, and GP41 is shown, GP41 is shown in pink or salmon color. And the fusion peptide is shown here is in red. It's just like a pigtail. On the right-hand side is one of the broadly neutralizing antibodies against fusion peptide, VRC34, bound to the GP140 trimer. So VRC34 has been shown to neutralize approximately 50% of 208 strains. And in addition to VRC34, PGT151 and ACS202 neutralizing antibodies can have a neutralizing breadth of about 66% and 45% respectively. So our goal is to try to induce similar kind of antibodies using vaccines. So much of the efforts to induce broadly neutralizing antibodies against fusion peptide has been done at the Vaccine Research Center at the NIH. And they have used fusion peptide conjugated to KLH or tetanus toxoid heavy chain. In addition, they have used SOSIP, which is a stabilized trimer from BG505 strain, as well as a consensus clay C trimer. And they have used guinea pigs, non-human primates, as well as mice. So in this panel shows many different vaccine strategies they have used. And as you can see, they have immunized with many different immunogens. The bottom line is that it is possible to elicit neutralizing antibodies. And they have isolated VFP16 and VFP20 from mice, which have relatively low breath. They also have isolated DFPH from non-human primates, which has a quite respective 59% neutralizing breath. Unfortunately, the problem is that the potency of these antibodies are low, and the serum titer is very low with ID50 of about 20 to 100. So we have been trying to figure out how to increase the potency and how to elicit stronger antibody titers against the fusion peptide. So when we began our work a few years ago, we started out with a very simplistic immunogens. So on the left-hand side is a immunogen that we just fused the fusion peptide to a pan-DR epitope, a helper T-cell epitope. And so we used that as a priming immunogen. And for the boosting immunogen, we used the original SOSIP GP140 trimer from the BG505 strain. And for detecting antibodies against the FP, we just used the biotinylated fusion peptide. So one of the vaccine studies we have conducted, we want to create novel vaccine strategies to focus immune responses against the fusion peptide. So one group we did is a typical vaccine strategy where you can see the, okay, there you go. It's a typical prime boost vaccine strategy. So we prime twice with FP Padre C and then boosted with GP140 twice. We're all with 5 micrograms. In group 2 animals, we immunized primed once with FP Padre C with 5 micrograms. And then we boosted 3 times with the GP140, but in increasing amounts. So we started immunizing with 0.5 micrograms, and then 1 microgram and 5 micrograms. In group 3, it's the same as in group 2, except that we included the priming immunogen, FP Padre C, for the 2nd, 3rd, and 4th immunization, but in decreasing amounts. Okay? So hypothesis was that by including FP Padre C as a boosting immunogen, we wanted anamnestic response against FP to target the FP on GP140. So we call this IPAS-RAM vaccine strategy, which stands for incremental phased antigenic stimulation for rapid antibody maturation. So when we measured antibody responses against FP as well as GP140, and for group number 1, we were able to detect antibodies against FP as well as entire GP140 only after the 4th immunization. In group 2, we did not detect any antibody responses against FP, which kind of makes sense because we only immunized with FP only once. And we were able to detect antibodies against GP140 only after 4th immunization, but it was much stronger than group 1. And in group 3, which was very surprising, is that we were able to detect antibody responses against FP only after 2nd immunization. So by including FP Padre C in addition to GP140, we were able to direct strong antibody responses against FP. And then after 3rd immunization, which included with the GP140, and FP positive C, the antibody responses against FP was much stronger. Okay? And seeing that, we were very excited, and we tried to evaluate neutralizing antibodies against the virus, BG505. But unfortunately, we did not see any neutralizing antibodies against BG505 wild type. But we did detect neutralizing antibodies against BG505 delta 611 mutant, which eliminates glycosylation site at position 611. And that's been shown to facilitate detection of neutralizing antibodies against fusion peptide. So, and we were able to detect neutralizing antibodies only in group 3. So, what this shows is that vaccine regimen can have profound effect on antibody responses. So what I'm showing you here is superimposed crystal structure or cryo-EM structures of 4 different neutralizing antibodies bound to triameric GP140 structure, except that I'm only showing GP140 portion. And panel B, I'm showing the same image without the broadly neutralizing antibodies. And panel C, I'm showing the enlarged view of the fusion peptide only. And panel D shows a perpendicular side view of panel C. So what this shows is that the fusion peptide is highly flexible. So there are many different ways of inducing neutralizing antibodies against fusion peptide. And the problem is, and the problem we have to solve is, how do you elicit antibodies against highly flexible peptide? So one strategy is just immunize with a free peptide, which we have done. And unfortunately, in those cases, because the peptide is flexible, you make antibodies against many different shapes. And unfortunately, most of those are not identical to what's on the virus. So they are either low neutralizing or no neutralizing. Another way to elicit antibodies is engraft the peptide onto a scaffolding protein to make it have a very stable, rigid epitope structure. Unfortunately, when you immunize with such an immunogen, you elicit much of the antibody responses towards the scaffolding, and you elicit antibodies against off-target epitope, which will not have neutralizing activity. So the solution we come up with is a vaccine strategy we call antibody-stabilized epitope presentation vaccine strategy, or ASAP vaccine strategy. And the hypothesis goes like this. So you bind a peptide to the neutralizing antibody, and then you immunize. So that, and you get antibody responses against the non-neutralizing phase, or the phase opposite of the neutralizing phase, and select for high affinity antibodies to the non-neutralizing phase, and use that to form a complex with a peptide to present the neutralizing phase of the peptide in open. And so the hope is to elicit antibodies that were similar to the original antibody, and we are hoping that would have a good neutralizing activity. So to generate such an immunogen, we actually generate a fusion protein. So we fuse the fusion peptide to the single chain Fv of VRC34. And to make sure that the structure of the peptide was correct, we crystallize it and we solve the crystal structure of this fusion protein. And so here is the heavy chain of the VRC34, here's the light chain, and here's the fusion peptide. And the structure is actually identical to the original fusion peptide structure found on GP140 and bound to VRC34. So we were very excited to use that. Now, the problem we actually saw was that the structure of the VRC30, the fusion peptide, was very low profile. So it's very, because there are many glycines and alanines which does not protrude out. So we thought, well, what if we modify the peptides so that we have side chains that are larger than glycine and alanine to have that fusion peptide protrude out of the VRC34 surface. And we found one a mutant version, and it bound to VRC34 to equal affinity as the VRC34. So while we were doing this, we actually found that fortuitously that if we immunize mice with MuFP single-chain Fe and then boost with BG505, GP140, we could elicit neutralizing antibodies quite potently. And so at that time, we had this data that I just showed you, that we were able to induce neutralizing antibodies against BG506 delta 611 with ID50 of about 30. So we thought, well, what if we immunize these mice with MuFP single-chain Fe? So we immunized with MuFP group 3 animals, 3 of the group 3 animals with MuFP single-chain Fe, 5 micrograms. We happened to use ASO4 as an adjuvant intramuscularly. Unfortunately, we got nothing. We then boosted again with another dose of MuFP single-chain Fe using aluminum hydroxide and immunized with intraperitoneally. And we got a little better, but not much. Then we thought, well, let's just immunize with a GP140 again. So that's when we got this result. So by immunizing with 5 microgram of GP140, we were able to induce high levels of neutralizing antibodies against BG505 delta 611 with ID50 about 1,000, which is more than 30-fold higher than what we had gotten before. More exciting, it was that we were able to neutralize the wild type with ID50 of about 30. Then we said, well, let's just immunize with GP140 again, but at a higher dose. So we immunized with a 25 micrograms, and what we got was much higher levels of neutralizing antibodies against delta 611 with ID50 of about 10,000, and then we also were able to introduce neutralize the wild type BG505 with ID50 of about 2,000. So we were very excited, and so we looked for whether the neutralizing antibody had breadth. So we did a neutralization assay with using pseudoviruses with Q23, SF162, BAL, and BI369. Unfortunately, none of them were neutralized. So then we did a structural bioinformatics analysis to see what the reason was, and we were able to show that and then we saw that there were various mutations in this region where the VRC34 antibody bound, and we saw that the BI369 had the highest similarity to BG505 strain, and it differed by only 3 amino acid positions. So we mutated D to E or N to S or a combination of both, and then we were able to restore neutralization sensitivity. So we know where the target was. Now, interestingly, for vaccine development, we figured out that 97% of all HIV-1 isolate had N241, which adds glycosylation site. So And we think that's the reason why we were not able to induce the broadly neutralizing antibodies, despite the fact that the neutralizing antibody titer against BG505 was high. So to better characterize the neutralizing activity, we decided to generate monoclonal antibodies. And so we used a typical traditional hybridoma method to isolate neutralizing antibodies that were able to bind to both the fusion peptide as well as the GP140 trimer. And we isolated 2 hybridomas, and so far we were able to clone both heavy and light chains of 5e6, but only a heavy chain of 74. 5e6 and 74 are clonally related with 94% identity for the heavy chain. And then we noticed that 5e6 is related to monoclonal antibodies previously isolated from mice by NIH Vaccine Research Center, with a heavy chain is about 80% identical to VFP16 and VFP20. And the light chain is about 87.5% and 85.7% identical to those neutralizing antibodies. We did not have time to determine the crystal structure of the antibody, but we used AlphaFold 2 to predict the structure of the 5e6 monoclonal antibody bound to the fusion peptide. And what we saw was that the structure was very similar to the FP protein bound to VFP16 and VFP20. So take-home messages is that, first of all, a vaccine against HIV-1 must elicit broadly neutralizing antibodies for it to be effective. Despite lower breadth, fusion peptide is a viable vaccine target. Vaccine regimen is just as important as immunogens for eliciting antibody responses. IPASRAM vaccine strategy is better than typical prime boost vaccine approaches. MuFP single-chain FP needs to be better characterized for its role in inducing FP-specific neutralizing antibodies. We think we know what's going on, but we need additional studies to confirm. BG505, GP140 might not be the best immunogen for eliciting FP-directed broadly neutralizing antibodies because of the fact that it does not have glycosylation site at position 241. We do need better priming immunogens to elicit broadly neutralizing antibodies FP, and we think we have figured out what is needed, so we are working on those. And we certainly need to streamline vaccine regimen to reduce the number of immunizations. I'd like to thank the people who worked on it most, Ling Niu and Vikram Srivastava. And this work is being continued from the neo-vaccine side by Santanu Ray Chaudhry and Raymond Larson. I'd like to thank especially the funding from the NIH, NIAID, their SBIR contract awards. Neo-vaccine has recently been awarded a phase 2 SBIR contract to further develop vaccine targeting the fusion peptide. And with that, I'll end. Thank you.
Okay, I will introduce today's last speaker, and also he is the co-organizer of this whole event. Professor Doko Kim. He is a professor of biomedical engineering and medicine, and the director of the Center for Microphysiological Systems. Dr. Kim is a fellow of the American Institute for Medical and Biological Engineering, the Royal Society of Chemistry, and the American Heart Association. His research focuses on the human microphysiological system for disease modeling, drug development, and space biology. Okay, Dr. Kim, please.
Yeah, thank you so much for organizing this conference, Professor Joe, and I'm going to talk about some of our microphysical system work for disease modeling and drug development and space biology. I've been working to minimize number of slides from my 45-minute seminar talk to the 50-minute slides. I try to highlight some of our relevant work. So this is financial disclosure slide. I am scientific founder of the Curie Bio and serving on advisory board for a few biological company. And this is an overview of my laboratory research. My laboratory research focuses on development of the microphysical model of the human organ. We utilize the state-of-the-art multi-fabrication techniques such as microfabricated biomaterials, microfluidic, and 3D bioprinting to create a more predictive functional human tissue by combining the human stem cell and tissue engine technology. We apply this human microphysical system for cancer and cardiovascular and neurological disease study. Today, for the sake of time, I'm going to mostly talk about our human iPS-derived cardiovascular microphysical system for several applications. Some of the speakers already introduced why we care about this human microphysical system. As you know, unfortunately, drug discovery and development is becoming slower and more expensive. And some of you probably heard about the Moore's law in the IT and semiconductor industry. So every other year we have the double, the transistor density. However, unfortunately, ironically, in the biopharmaceutical industry, there is an opposite law called the Ehrum's law. So every 9 years, we have a significant lower number of the FDA-approved drug. One of the major reasons this happens is the modern mismatching problem. So we care about the human disease biology and human disease treatment. We are using the animal-based preclinical model. So to address this issue, NIH identified this is a major area of interest and created a roadmap. Since 2012, they developed the safety pharmacology and drug efficacy, and more recently, clinical trial chip for the precision medicine program to change the way human disease therapy has been developed. Recently, a number of the big pharma participated in this NIH tissue consortium, and this is some of the major research group involved in this more than 10-year-old NIH tissue program. So tissue chip consortium 1.0 is concerned tissue chip development for the safety pharmacology study. Later, tissue chip 2.0, a number of studies has been done for disease modeling and efficacy testing. And ongoing NIH-funded program, clinical trial chip for precision medicine, our scientific team is one of the awarded team, and we are trying to utilize the patient-specific and organ-specific model for precision medicine development. So based on this, all the hard work and NIH and scientific community effort, in 2022, December, in United States, President Biden endorsed the FDA Modernization Act 2.0. So based on this, we can use the alternative method without including the animal testing data. We can use the in silico or human MPS data for IND filing. So there's a number of the implications associated with this FDA Modernization Act 2.0. So there's a lot of promise. However, there are a lot of the scientific and technical challenges in this area. So I summarize the 3 major technology challenges. First, most of the commercially available and academically published microphysics system has a low throughput, n = 1 study. So we need to improve the throughput so that this can be utilized for industrial application. Second, we concern with the living tissue and living organ, so we should be able to have the longitudinal phenotypic assessment using those living organoid and living tissue chip without fixing tissue and organoid just to study the static biology. So we should be able to integrate the biosensor, allow us to have the long-term monitoring of the drug efficacy and disease progression. It's just like the human patient test in clinic. So finally, most of the fabricated microfluidic system and organoid has been manually produced. So there is lack of the producibility, batch by batch variation. So we need to make a more robust, scalable, automated manufacturing method. So in order to address this, my lab has been focusing to develop the high throughput biosensor integrated automated tissue platform. So this is one of our developed platform. So we have the 24 engineered 3D hard tissue, and one of the we have an embedded magnet, and this is a fixed rigid post. So this is a human iPS derived cardiac tissue. So whenever cardiac muscle cell and tissue twitch, we can measure the magnetic field change based on the movement of this embedded magnet. So previously, scientific group measured the displacement based on this post deflection using the live cell microscopy. It's very expensive and bulky and also low throughput. But here we can measure the engineered hard tissue functional contractile kinetics over time in non-invasive manner or from the 24 tissue. So this technology has been commercialized through our startup company, Curie Bio. It's commercially available for drug discovery application. It's called the ManTray. So in addition to the force measurement, we can also assess the frequency and rate of contraction, relaxation, and number of the other functional kinetic parameters in real time. So if we have this measurement for a couple of minutes, we open store about terabyte data. So we store this in the cloud space to run some machine learning or big data processing algorithm. So tissue consists of the biological cells and extracellular matrix scaffold. As one of the speakers introduced, our lab also utilized the cardiac tissue derived cellular matrices. But we combine this with reduced graphene oxide so that we can tune the mechanical and electrical property of this hybrid scaffold for advanced cardiac maturization. So this is a structural property of our hybrid scaffold, very similar to the native myocardial structural property. And when we create the engineered human tissue based on the collagen scaffold or DCM RGO, our proposed hybrid scaffold, we demonstrate within 2 weeks, we have significantly enhanced cardiac maturation in terms of contractile force generation and also electrophysiological function, other gene expression. So we use this as our established protocol and apply this platform for disease biology study and phenotypic drug screening. So as I mentioned, the 24 medium throughput platform has been commercialized through the Curie Bio, and this has been utilized by the more than 20 pharmaceutical companies for drug discovery, particularly for the muscular dystrophy therapy. And our Johns Hopkins group recently scaled this 24 medium throughput platform to the 96 high throughput platform by optimizing this micropost aspect ratio and also magnetic field interference between these two post system. So I will skip some of the slides for the sake of time. So for 24 tissue chip fabrication, my postdoctoral trainee or graduate student can make the 24 tissue without any problem. But 96, it takes 30 minutes to sometimes hour. So none of the tissue is actually identical, and it's not reproducible. So we applied a very simple XYZ liquid handle robot to automate this 90 tissue chip fabrication. So it turns out, we can complete the 90 tissue chip fabrication within 6 minutes. So we save tissue fabrication time by 5 to 10 times, and more than 90% in success rate. So whenever we have the 96 tissue chip fabrication for high throughput drug testing, we utilize this established modular tissue chip fabrication process using the liquid handle robot. So using this kind of platform technology, which is more high throughput and biosensor integrated and automated, we apply this for our disease modeling and drug discovery application. So we primarily target the Duchenne muscular dystrophy, and this is X-linked monogenic disease muscular disorder. Since this is X-linked, only boy has this problem. 1 out of the 3,500 boys. So although this is considered a rare disease, there are huge population, and there's no cure available. And since this disease is very severe, a number of the big pharma aggressively developing the gene therapy and other targeted therapy. And most of patients died due to the breathing problem or heart failure. So in order to develop the potential therapy, we propose to develop the patient-derived human-relevant disease model, and we can screen a number of different therapies using our high-throughput 24i96. And we can develop both cardiac and skeletal muscle tissue chip, which is relevant to this particular disease symptom. So our group have obtained more than 20 iPSC from the Duchenne muscular dystrophy patient and also Becker muscular dystrophy patient, which is mild form of the Duchenne muscular dystrophy. We partner with Boehringer Ingelheim, which developed the TRP6 inhibitor for DMD. It's currently under clinical trial phase 1. So just like the clinical trial, instead of the actual patient in clinic, we utilize the patient-derived human disease model in the laboratory and test drug safety and drug efficacy in the small scale, 3 isogenic pair of the patient-derived iPS model, and phase 3 whole 21 patient-derived model for large-scale drug efficacy. So this was funded by the NIH Clinical Challenge Chip for Precision Medicine program. So there was a go-no-go criteria in 2 years. So we first had to demonstrate our proposed disease model can actually recapitulate the human patient symptom. So here we developed both engineered heart tissue and engineered skeletal muscle tissue. As you can see, compared with the wild type tissue, dystrophic cardiac tissue and skeletal muscle tissue has a significantly lower force generation, which is very similar to the actual patient symptom. And since this is monogenic disease disorder, many big pharma utilize the AAV minidystrophin, but this is a very big protein, so transcript form of the dystrophin has been targeted. Pfizer and Sarepta is running the phase 3 trial using the AAV minidystrophin. So we tested Pfizer and Sarepta, their AAV minidystrophin formulation, and found that the Pfizer minidystrophin has some improvement in the cardiac function when we changed the dose of this AAV. Unfortunately, Pfizer recently stopped the phase 3 clinical trial because one of the patients from phase 2 died of the heart failure. So we need further investigation using these types of the in vitro disease model. And here we compare the Pfizer and Sarepta AAV therapy and comparative analysis. So using our high throughput platform, we can test with the other types of the novel therapy for DMD therapy application. So for next few minutes, I'm going to introduce another application of the microphysical system for space biology and medicine. So NIH partnered with NASA and created the tissue chip in space program. So 9 scientific team has been awarded, and we was one of the team to study effect of the microgravity on the heart function in space. So it's very well known that space microenvironment accelerate the aging, and many space astronauts suffer from this microgravity induced diseases. So this is our experiment design. So we differentiate iPS into the cardiomyocyte and cardiac fibroblast, and then create the multicellular microfabricated tissue. And some of the engineered heart tissue we send into the International Space Station, and others stay in the laboratory and serve as a ground control. And we measure the contractile force in real time from both space and Earth. And once this tissue flown back to the Earth, and we collect the sample and run the structural analysis and also RNA sequencing. So we had to redesign our tissue device, because in space, medium can spill out of the multi-well plate. So we had to make the tightly sealed 6-well tissue chamber, and we use this as a modular unit. So in this device, we have the 8 of this 6 tissue chamber. So we have 48 heart tissue in this single device. So we actually send this to the SpaceX 21 in March 2020, and then we analyzed the tissue function in space versus the Earth. I mean, this is space astronaut Jessica Meyer, who changed media once a week using our device. This is a protein device inside the glove box, which you can see from the NASA website. So this is our experiment data. So as you can see, over time, space flight sample gradually decreased the force. So there's a heart failing, and another main function of the heart is electrophysiology. And we found that in space, this space tissue sample display the irregular heartbeat. Delta BI is beat interval variation. So if heartbeat is regular, then it's supposed to be nearly zero. So ground tissue, you see the very consistent, nearly zero beat interval variation. However, space flight sample on day 19, there's a peak in the beat interval variation. So there is some kind of the arrhythmia in space versus the Earth. But as this tissue flown back to the Earth, there is a gradual decrease in this delta BI. There's some self-adapting mechanism in the heart tissue. So we collected this tissue from the Kennedy Space Center and run the electron microscope analysis. Normal heart, there is no repeat accumulation, but in this space tissue sample, you see a number of the repeat accumulation, which is very unusual. And there is also mitochondrial defect and mitophagy from the space tissue sample versus the ground control. Also, the myofibrillary and sarcomere structure is broken in the space tissue sample versus the ground control. And this can also actually support our functional change in space, decrease in the heart force and arrhythmia. And based on the RNA sequencing analysis, we found downregulation of the calcium signaling and hypertrophy and cardiogenesis, and upregulation of the heart failure and metabolism and inflammation. This also further support our phenotypic changes. And we identified the key gene involved in the microgravity-induced cardiac dysfunction. So in the heart, contractility, ion channel, mitochondrial, and oxidative stress is important. And this pink color indicates the space flight relevant gene expression. You see that there's a significant decrease in gene expression in terms of contractility, ion channel, and mitochondrial oxidative stress. It's not just a few-fold changes. It's more than 10, sometimes like 100-fold changes. So based on this successful first space flight experiment, we was allowed to launch our second space flight sample for drug testing to attenuate the microgravity-induced cardiac dysfunction. So since we have very short time window, we was not able to use any novel therapy, but we utilized the FDA-approved drug. So we tested some of the potential FDA-approved drug using the random point system. So we adapted this from the Airbus and used the same chamber and same sensor and same tissue. Interestingly, this simulated microgravity setup actually predict this microgravity-induced cardiac dysfunction. So in week 3, there was a significant decrease in the contractile force. Also, there's arrhythmia. So we tested with 8 different drugs using this random point system, and then narrow down to 3. So in March 2023, when we sent our second space tissue sample, we tested with these 3 drugs and found that one of the drugs, which I'm not able to disclose due to the IP issue, and showed a significant promising improvement in this microgravity-induced cardiac dysfunction. So our first space flight study has been recently accepted with the minor revision in the PNS, but our second space flight sample, we still analyzing the post-flight sample, RNA sequencing, things like that, but has been highlighted by some of the mass media. So this is a summary slide about some implication in the microphysical system. I think this is one of the most interdisciplinary area involving the biofabrication and stem cell technology and AI and big data science. So major keyword in this emerging field is the standardization, automation, and miniaturization. So as I mentioned, the NIH layout 12, 13-year funded program, but they recently announced another next decade-long program to integrate the microphysical system with the other new approach methodology like digital twin and AI big data and in silico combination with the non-animal and human-based disease modeling. So in order to tackle this significant problem, I team up with a number of the Hawkins investigators and recently established the Hawkins Center for Microphysical System. So we identified the 4 major technology areas shown here, and also the 5 disease areas. And combined with the data science and AI approaches, Hawkins, we have a huge patient population. So we are focusing on the precision medicine approaches using this enabling platform technology. So I'd like to thanks to my group member and funding sources, and happy to answer any question during the Q&A session. Yeah, thank you.
Hello everyone, let's start. Shall we start? Thanks so much for your attention to these outstanding lectures by this great scientist. So this now we receive your question, but before receiving question, we have some question for the each speaker. So the question is, how about application of AI in your field? So how to apply AI technology in your field for cost-effective, decrease cost of your technology? So everyone, please kindly speak about that. Yeah. Please. Thank you so much.
In my opinion, AI is good, but you have to be very careful.
it's a hype. So garbage in, garbage out. So if you don't have a good data, the AI will deliver you garbage. So you need to have a real data. AI without real data is totally useless. It's totally hype. So you have to really, really be careful. I'm not ignoring AI. I really appreciate AI. But however, I like to emphasize that if you don't have a good data, it's a garbage in, garbage out.
Thank you so much. Thank you.
And I think from my perspective, studying acute respiratory disease, it's really understanding that treatment options that begin earlier are the most successful because you only have a small window of time. And so where the AI technology is fantastically interesting to us is to try to identify very early markers that maybe aren't even associated with the disease pathology, but are those precursor signals that suggest the person may be more disposed to severe disease, and then understanding how you can then implement those treatments at those early factors. So before a person is really sick, can you tell this person needs particular care? Can you put them on oxygen supplementation? Can you put them on other things to prevent disease? And again, that preventing disease is something that is important to me from a public health standpoint.
Thank you so much. Yeah, thank you.
Yeah, so for our work, AI is mostly used as a technological advantage. So just to give an indication, any time-lapse experiment we do, typically we visualize approximately 100 organoids in one experiment, and this yields about a data set of 500 gigabytes up to 1 terabyte. If you need to analyze this by hand, you need a year. So we use AI a lot for improved segmentation of nuclei, tracking of cells, and so really use that to our advantage. But also here, it is garbage in, it's garbage out. You need good training data. So the time investment is really needed to do this well. But it's definitely an improvement to what it was before. Yeah.
Thank you, Saskia. Yeah.
Well, yeah, I think it's similar. I think it's garbage in, garbage out, right? In the biofabrication field, the way the AI is now being integrated into most approaches is to accelerate the optimization process. In most of the printing process takes really long, and we don't want to repeat the failure. But again, if you don't have the right data, you're not able to use AI to help optimize it. So there's numerous amount of papers coming out now on the use of AI in bioprinting. But beyond that, I think there's a lot on AI, machine learning, but also a lot of digital twins coming out as well. Like how can we start setting up processes that are able to help low middle income countries, that you can have processes set up in more wealthy countries so that you don't have to reinvent the wheel for your low and middle income countries. I think that's one of the key priorities that the World Health Organization have set for bioprinting in general, and I think there's some policies coming out in the use of that as well, in implementing it into the field.
Great, thank you so much. Thank you.
I also agree with what other speakers have said. For aging, one of the best uses for AI is predicting your biological age. We kind of predict our age as a chronological age, so every year, 365 days, you age 1 year. But actually, your biological age is a little bit different. And there are a lot of aging clocks based on the DNA methylation. So everyone ages differently. So it could be a powerful tool, especially in Korea. Like in US, the genetic background and the population is so different. It's really difficult to apply. But in Korea, we have, at least we're one race, and also for Koreans, we do a physical checkup every year. So we have, medical system is much better than US here because of healthcare. So AI could be a powerful tool to study aging and also for rejuvenation as well. So that's my opinion.
Okay, thank you so much.
In developmental biology, and also I do in our lab like drug screening platform, and using the AI, we predict how cell differentiate properly, or we can kind of anticipate some disease differentiation. So I think we can save time by predicting using the AI. That's kind of what we
Thank you so much.
use.
Thank you.
Okay, so as you've heard my talk, I'm engaged in the vaccine development and in focusing primarily on humoral immunity. And antibody response is largely based on a random process of your immune response, and the antibody maturation process is largely a random process. And I don't think there is any AI that can predict immune response, but I think AI can be helpful, or at least computer can be helpful in terms of immunogen design. In the past, to design immunogens, you would have to first express the protein and determine if their crystal structure or cryo-EM structure. But as we're trying to design smaller immunogens, I think the AlphaFold has been very important in helping me design new immunogens that could potentially elicit the type of immune responses that I like to get.
Thank you so much.
I agree with what Dr. Lukri mentioned earlier. It's very important to establish the quality, big data in order to make this AI enable drug discovery and other research meaningful. So there's a lot of the patient data available. So that's why medical doctor and clinic, they emphasize the AI and data science. But in the emerging field of the human organoid or human microbiology system, enable the drug discovery, we need to integrate the biosensor, real-time biosensor, which allows us to collect the quality big data, which is quite missing in the field of the microbiology system. So in order to make the AI-based drug discovery or MPS research, I want to emphasize the real-time predictive biosensor integration is very important to establish the quality in the big data.
Yeah. Thank you so much. So now we could receive questions from the audience. So please, if you have any questions for any of these speakers, so please, we'll be happy to receive your question. So go ahead, please.
Okay, so in terms of the AI, is the previous question. I think is how do we define the AI? People sometimes call the AI and the big data is differently. So when it said is the developmental biology, especially is one of the complicated field because the system is changing as time and it develops. So the biology is diversity, but developed biology is more complicated about that. So we deal with lots of big data. So when you get some maybe top 5% or top 0.5% or top 0.01% or something, dealing with the big data, is we need a computer system because they calculate better, provide some accurate data set. But AI is something beyond that. As with all the data, they decide, they select something and propose something. So I think AI is like personal. All the person is people of AI is a different personality. I'm using the AI of maybe the people one. Or some people using the computer as they get the answer, but the AI is like person 1355 or something. So it's when you get the AI answer, there is some personal based on their learning experience. They provide one of the best answer about that. So the data could be something not fully formed. So that data is not complete. But based on that, they try to provide based on their own experience. So after we got the answer from the AI, we should check. That is important. We should verify about that. I think that is the best, most important thing we need to do that. So, and I think the big data and AI could be slightly different, but I'm very thankful to get the help of the AI. They try to deal with lots of data and they try to summarize or suggest something, and I get the advice from my colleague about my research, but I cannot trust that 100% about that. And one question is, I try to ask a question to the Dr. Zhang at the Emory University. Even we try to compare the cells, some gene expression on some specific time point. It's very complicated. About 20,000 genes, how they express their level is really, but you dealing with two different individuals. You connected them, and there will lots of the signal will be communicated. And after that, you try to identify which factor is making them younger or something. I think the background error or something is too high to identify some very significant one signal. Do you think it's possible? Because it's too complicated. And did you identify one of the some strong signal? Maybe that will be the first signal or something.
Okay, so that's a very good question. Actually, so the reason that we went with the 3D microphysiological system is because of that reason. So I believe organ on a chip and 3D MPS system is a functional unit. So there are many things. It's multifactorial how aging process occurs, but we can narrow down to one function. Like Professor Kim presented, his model is for contraction. That's one function. Our model is myogenesis. So our endpoint is always proliferation and differentiation of the stem cell, the muscle stem cell into myotube. So if you're just looking at myogenesis as an endpoint, we could identify factors that are playing a role in myogenesis, which is one of the rejuvenation effect. So if you combine all of that with the help of AI, we could kind of have a better understanding of how the aging process occurs in the muscle. And if you expand that to other organs, how the cardiomyocyte work, how the liver works, how the other works. So we don't really need to have a big picture, and we could just start from, it's sort of like a reverse engineering. So we start from the simple system and expand it as we go. And over time, it's going to change. So initially, we wanted to start with the most simple system, but we're now expanding. And this isn't like one lab task. We need to have a collaboration with other investigators as well to really tackle aging.
Thank you so much, Dr. Zhang. So please try to ask questions. So yeah, please go ahead.
Maybe this can be quite a specific topic. I have a question to Professor Luke Lee from Harvard University. Probably you don't have enough time to explain it, but you showed us a lot of devices. So how did you detect the biomolecule or the bio event in a cell? And the other question is that could you explain about the QBT more than QBT?
Okay, let me start with the QBET. QBET is a quantum biological electron transfer. We can detect electron transfer while during the cell death, because you can use a plasmonic to capture cytochrome C oxidation reduction in real time. There's no such tool, but it's very easy to adapt with the spectrometer and then dark field microscope. Then you can capture this real-time electron transfer from cytochrome C. That's QBET. And then another question is, you talk about sensing for molecular diagnostic or sensing in organoid?
Actually, both of them.
Oh, okay. First, sensing for molecular diagnostic, we are using real-time PCR on chip. So we can finish the PCR in less than 3.5 minutes. Usually PCR take 1 hour here on chip, we are using, again, plasmonic heating and cooling. So you can take advantage of the photonic PCR cycle. So you use the light on and off to heat it up and cool it down, or the photonic nano-optical antenna. So you can take advantage of this ultra-fast photonic PCR in addition to protein detection, protein biomarker detection, but RNA detection is very important for infectious disease. So it is important to think about array of different pathogen detection on chip so that you can envision. Actually, this technology is available. It's a matter of manufacturing. Okay, so many people follow up our work on photonic PCR. Last almost 5 years, not only from America, Canada, Korea, China. So that shows that it is possible. So you can take advantage of the photonic PCR on chip for infectious disease, or even imagine if you worry about cancer, early detection of the cancer at home. So it's possible. The one, the FDA approved the home diagnostics device that I show you, that my student made the company, Lucira. They use actually a more simpler nucleic acid amplification called LAMP, isothermal amplification. So even LAMP can be accomplished within 30 minutes. Okay, in terms of the detection, as Professor Toko Kim mentioned, that it is really a good time to think about how to capture signal, not only molecular signal, for example, through the exosome. We can detect, again, protein biomarker from exosome and RNA or DNA marker from exosome, because we are collecting the released exosome from organoid on chip, as well as detecting real-time detection of the brain wave without touching the cell. So brain wave can be monitored and see how different part of the brain wave, is it beta or gamma wave, is changing due to the toxicity or due to the different inflammation factor. You can observe. So that's one of the non-invasive detection method. But this method can be used for cardiology. The EKG is a similar concept. As you know, that our cell has the same gene, whether is it from brain cell or cardiac cell or even kidney cell. You have to think about that. Every cell has an ion channel. That means you can generate different electrophysiological signals. So you can think about detecting different organ, even electrophysiologically. But same time, you can combine molecular expression through exosome or from secreted molecule, or same time, non-invasive electrophysiological expression can be detected in real time. So this kind of precision data is very important for AI to make, good, smart decision with the pattern recognition.
Thank you.
Thank you so much. So please, any question more? Any more question, please? Yeah. Oh, so please, yeah.
Actually, I'm apart from the AI system, but I just used the ChatGPT and asked what is the next pandemic. He told the highly pathogenic influenza virus will be the next. So, as a virologist, I think I agree that. Dr. Pezos, do you also think next pandemic will be the highly pathogenic AIV? And also, if then, you told about the antigen changing, especially neuraminidase changes, alter the many pathogenicity and like that. So, Tamiflu will be working on the next pandemic?
Yeah, no, so I guess my personal thinking would be, yes, influenza would certainly be a high candidate for being the next pandemic. Of course, I thought that before COVID-19 as well, and we can see how that worked out too. I think that there are, very important similarities among all of the pandemics that we've seen over the course of the last, let's say, 120 years or so. And for me, the interesting part is it's almost not as important which of the viruses causes a pandemic, but again, from the public health, from the prevention standpoint, it's understanding where's the weakness that allows these viruses, right, to enter the human population. And if we can find and move back in an approach to try to minimize that aspect of it, then, we can minimize the risk of a flu pandemic, a coronavirus pandemic, or an arenavirus pandemic, right? By being able to think about the processes that are going into place and minimizing the risk there. And I do think, Dr. Cho's work on vaccine designs is, I think, another area that's really important, because there certainly are ways that we can make better vaccines that target more influenza virus strains, that target more coronavirus strains, just like you're trying to do with HIV strains. And I think that's another place where I think we can make a lot of progress, because right now, we're focusing very much on what works against seasonal viruses in human populations, looking at the short-term benefits. We need to take a larger approach and try to understand the long-term benefits of vaccine design, and really think about what's the best strategy to generate those long-term immunities. And things like the neuraminidase in influenza is a perfect example. The influenza vaccines don't generate neuraminidase antibodies, but many studies have shown that the neuraminidase antibodies that are present in people can modulate disease. And currently in the US, we have this H5N1 avian influenza virus. And when people do animal experiments in immunologically naive animals, the virus is very, very dangerous and very, very lethal. But if you put a little bit of neuraminidase immunity in those animals, suddenly the disease severity drops significantly. So I think understanding how some of these alternate targets for vaccination might also help minimize severe disease severity is an important area to look at.
Thank you so much, Andrew. So please try to ask more questions, please. Yeah. Okay.
So I do have a couple questions for Dr. Lee, specifically regarding to the MAP system, where you described a perfusion barrier that facilitate the circulation of the necessary factors for the brain organoid. So I was wondering, such perfusion barrier, is that something that could mimic the effect of a blood-brain barrier that we expected in physiological situations?
That's a very good question. Theoretically, you can do that, but if you really want to make a much better mimicking the biological system, it's good to put the barrier cell. So you can add the cell on the perfusion layers. Actually, many people copy this perfusion channel and culture the cell, and they claim it's BBB chip. So it's kind of good extension. So when you develop this kind of design, many people come out with more biological adaptation and application. So you can find BBB on a chip has this kind of structure.
Okay. And one more question specifically on the brain organoid itself. So you mentioned that you collect EV through media collection, daily collection of the media. So I was wondering, how long could such brain organoid be maintained? And if it can be maintained for a long term, or specifically, how many days do you collect the EV for as a set of experiment? And then if it could be allowed for a long-term collection of EV just within that specific organoid, then is there, could there be an aging effect from day to day?
Oh, absolutely. So usually for the brain organoid, it takes really long time. So we check, every 10 days and then see whether we can continue to do experiment even after 3 months. Okay. We did experiment up to 90 days. But it's very expensive. It's IPS. Okay. So we didn't make a record, but we just stopped at 90 days. But theoretically, you can continue to culture. But it costs a lot of money, especially IPS.
Okay.
Unless you have a reason to do that, I'm not sure whether you want to say, okay, we culture 1,000 days and make a record.
Yeah, because it's more like daily culturing for like regular 2D cells. You join like even just from passage to passage, we see an aging effect from the early passage to later passages. So there are also reported effect of like how EVs secreted by the aged cells could be different from the EVs secreted by the younger cells. So that's why the question comes that
I see. Those kind of experiments need to be done more systematically. And I really recommend you to challenge you on this because it's a very fascinating area. I'm not a developmental biologist, but if I'm your age, I'll act like a developmental biologist because even though I study biophysics and engineering, but I realized that developmental biology is a fascinating field and there are so many things to study. So using this tool, this is your tool. We are nothing but servant of you if you're a biologist. And we will provide this tool to study more precisely with the flow. Instead of using flat petri dish-like, you can think about how to mimic our physiological system more effectively. I'm not saying that this chip is the perfect, but at least better than flat and statics, right?
Thank you. Thanks.
Thank you so much. So do you have any question, please? So any question? Oh, there are so many questions. Okay. So Professor, okay.
So I'm really honored to attend here, and I have one question for Professor Ashika. And have you ever mixed the two different types of cancer cells, and what's the result?
Yeah, thank you for this question. It's yes and no. So what we did is take the same cancer lines that we use, but we, in mouse models, enrich them for growing in the liver to get a more metastatic line of with the same oncogenic mutations. And we tried to do competition assays with this, and actually it didn't give a difference. So it told us that even though they have a different fitness in vivo, it doesn't mean that this is the same when competing with each other. So that's one example. And another example is using human colorectal cancer lines with a BRAF mutation, where this was on a collaboration basis, where we corrected the BRAF mutation and then mixed those. And here we instead saw a cooperation between the cells. So we didn't see competition, but we actually saw that the two different cell populations need each other to grow better. But yeah, you can do anything with these models. So it's a very cool idea to look at that as well. Yeah.
Thank you very much.
Thank you so much. Thank you. So next. Okay.
I have a question for Dr. Lim. Firstly, I wanted to thank you so much for your wonderful presentation. I wanted to ask you about your opinion on what restricts the current tissue engineering and regenerative medical strategies from us being able to make like large amounts of clinically relevant sized tissues or organs.
Thanks for that. I think that's kind of a very big question for everyone in the field. From a tissue engineering point, I think the major challenge or the question that we are facing at the moment is that why are we not yet able to make to grow organs in the lab that we can already transplant? I think that's a question that we all think is based on vascularization. So I mean, a big chunk of our research is looking at scaling up by building the vessels. And once you get the vessels right, you should be able to then integrate it with all kinds of tissues. Of course, we have avascular tissues in the body, but for a lot of the vascularized tissue, getting that scalar network right, and then we should be able to then scale that up. That's one part per se, but then the other part is once you do that, how do you implant it and it would integrate well with the body's native system? I think that's the real challenge in the moment. And with your question, I think I do want to bring a bit of awareness also with the media hype and a lot of misconception that is out there, where we're not yet bioprinting an organ per se. We can print a lot of things in specific shapes, but functionality is the important one. So you've seen a lot of papers where people print a human nose or print an ear, but to be fair, none of them are actually giving you the function that you can't smell things or you can't hear things. So I think another bigger misconception in the field is what are we trying to achieve? I think function is way more important than trying to just get the shape.
Thank you. We have one or two more questions. So please. Please.
Okay, so for the Dr. Zhou at the Iowa University, I totally have been forgot about the age because of the pandemic of the COVID. Nobody has talk about the age, but it has been already 40 years, and we have no vaccine. And do you think some RNA vaccine or different one can be the new vaccine for the age? And do you think what will be so hard to make the vaccine for the age?
Well, so first of all, I think the mRNA vaccine will be able to provide a new avenue of antigen delivery. So it could have an impact on development of AIDS vaccines. But I think the fundamental problem with HIV vaccine is that antigenic variation. And that is a difficult problem to solve, and I'm not sure whether mRNA vaccine would be able to allow to overcome that problem. Because the vaccine, I think the major difficulty is how do you target conserved epitopes that are critical for the neutralization. And as I mentioned before, immune response is by and large random process, and antibody focusing onto target that are highly conserved. There's no real way to control that by giving an antigen. So I think, what I'm trying to do, how I'm trying to solve that problem is by forcing the immune system to focus onto the critical conserved epitopes, like the fusion peptide, by modifying the formulation of the antigens, and the strategies that you use in addition to immunogen. So I hope that answers the question. But so I think the way the mRNA vaccine could help is by delivering multiple immunogens. But I think that has to be done in conjunction with the immunogen design as well as the regimen.
Thank you so much. Thank you. So now now time is running out. So anyone, you have a question? Okay, so last question. Yeah.
Hi. So I have a question for Dr. Seong Ri. So I see that when you do the in vitro model with the iPSCs, when you try to create the vascularized organoid, you use an approach where you fuse, if I'm seeing it correctly, that you fuse kind of a spheroid or an organoid of endothelial cells together with the cardiac organoid. How would you expect it to differentiate from if you induce endothelial cells with the cardiac lineage directly from one embryonic bodies instead of fusing these two together? Do you expect it to affect your readout? Yeah, that's my question.
Great question. Actually, we have two methods for vascularized cardiac organoids. One for, as you just saw, we differentiate endothelial cell cardiac separately, and we merge like a more spheroid type. And the other is we can also differentiate vascularized cardiac organoids from the iPS cell. And we kind of use, to optimize this model, we use the reporter line. So for example, red color with the endothelial cell and green color cardiomyocytes, so we can kind of optimize and for our in vitro model.
I see, thank you. Also, I have one last question for Dr. Kim regarding the space biology project. So I think when the heart is subjected to microgravity environment, one significant change is the change in preload and afterload. But then I think this model, the preload and afterload is mainly determined by the passive force exerted by the tissue post. So then even though we see a difference comparing the ground control and the spaceflight tissues, but then this preload and afterload is kind of not altered. So how would you, do you think there's a way that, or do we need to improve the model to fit this purpose, or how much would you trust the difference that you observe between the ground and space control, despite there's no kind of difference in terms of preload and afterload in the tissue model?
Thank you for good question. I have several different answers. My general answer would be we need to keep innovating the model system. In order to fully predict the human relevant physiologic response, I think let's say we need like a MPS version 10.0 or 11.0. I mean, we are in the era of the Windows 11, right? But at this point, we are using the MPS version 1.0 or 1.1. It's very early stage. So I just gave you some case study where we found something interesting, but of course we need to advance our model system to better predict the human space response. So that is my general question. More specific answer would be, it would be great we have opportunity for our third space flight experiment. At that time we can have a more fundamental scientific question. But as you know, it's very difficult to have this kind of space flight opportunity. So it would be more practical to utilize the simulated microgravity setup to ask this kind of specific biological question and see whether we can validate this in one of the future space flight experiment.
Thanks so much.
Thank you so much for your excellent question. So now time is already we behind the schedule. So thank you for the excellent question, and thank you for the speaker for effort to answer your question. And now we can close our session. And then thank you so much for coming and still until now and hearing the lectures. So thank you, and give big hand to the all speakers.